Excellent. Good afternoon. This is Thursday, August 27th, 2020, our second lecture of the cardiovascular system. Any questions before we get started today? All right. I want to address a couple quick things. I know we went through the materials you are responsible for in this class a little quickly because I've had some questions about that. So I did want to quickly go over a couple quick issues, some things that people had asked about. So let me go ahead and get to the right screen to be able to do that.
I did it. I'll just honestly leave that up in case you...
Yep, I would leave it up too. Oops, wrong button. So again, I know most of this is stuff that we went over in the introduction, but like I said, I appreciate that we went really fast through that. So I don't mind taking a little extra time to go over this again. There are primarily two main lab simulators you are going to be using in this class. The first is Labster and the second is PhysioX, although I purposely have given you an interactive physiology assignment in the first one. First module. Let's start by again going over this again, getting started stuff. Some of you, I'm in the process still of grading the Proctorio quizzes. As soon as I get them graded, I will publish the grades for that, and then that way you'll get a chance to see my comments. We'll talk about that a little bit more in the rest of the introduction, but I want to remind you, for those of you who weren't quite sure about the scanning, I do have these three examples of how to scan your area. One of the things that I want to point out, I know it looks really fast on these videos, but you have to remember that the video I get is sped up because if it takes you 4 hours to watch the exam, I can't sit here for 4 hours and watch every single student towards that. So it does speed it up a little bit. So you don't have to go as fast as those videos are. In fact, you should do the opposite. You should go slowly through them. This is where you get to our YouTube class for the lecture slides. I mean, the lecture outlines that are, the videos that are there. There's also someone asked a great question about the Science Success Center. I'm going to get a link to that directly, and I'll add that to the study tools for those of you who are interested in doing that. As I mentioned, here are the lab simulators. Most of the graded assignments are going to be either out of Labster or PhysioX. Labster is a program that the school pays for. And so it is an app basically that has been added to our Canvas shell. And so you are able to link to it directly from this. So when you click on this link, it takes you to the introduction to it. This pops it up in a new window. You can do that activity and it automatically saves your completed activity to your Canvas site. And remember, you can go to that Labster dashboard to actually see the activities you've done. And again, I haven't done any of these activities yet, so my dashboard is blank. But you access it through the modules. The other assignments are going to be the PhysioX primarily. These PhysioX we get through Pearson, through that Mastering A&P. So for this, remember, you have to go to the MyLab and Mastering. You have to open up your MyLab and Mastering. You have to go to the study area and you have to go to the PhysioX there. When you complete those activities, you will get a lab report. Save that lab report as a PDF, and then you're going to come back to your module, click on the link here for the PhysioX exercise 6, and you're going to submit that lab report here. There are 5 different activities. You're going to get 5 different lab reports. You can submit them all at once. Notice when you hit the submit, you can add more files, so you can submit as many as you want at once. Or you can do them one at a time. When you do them one at a time, I know it does look, it says like resubmit, and there's a fear that it overwrites your previous files. It doesn't overwrite your previous files. Whatever you submitted for before is still there, and I can still access it. So if you want to do one today and one tomorrow and one the next day and one the day after that, and turn them in each as you complete them, that is perfectly acceptable. All right. The third thing, and again, I've mostly put this in here as a carrot, as a reminder to you, is that the third lab simulator we have is the interactive physiology. Interactive physiology, as I mentioned, is a directed tutorial that is also in the Mastering A&P. And I can open that up and show these things to you guys again to remind you of that. Here, we do have a direct link to this activity because it is a tutorial through the Mastering A&P, and there's a PDF that needs to be filled out. But let's see what happens if I click on that. I don't know what happens if I do that. Ah, there we go, excellent. And it goes to the activity that you're responsible for. However, and I don't know, see, yeah, this won't let me do that. So yeah, interactive physiology is one of the tools, and there are many, many tutorials in that section. See, this just allows me to access this one with this link, but when you're in the study area and you go to interactive physiology, there's one for the cardiac conductive system, there's one for the cardiac action potentials, there's one for all sorts of different things. It is a great resource that you may not have used in 430 because it isn't emphasized as much, but in 431, we're gonna have a lot more physiology. So I strongly encourage you to take advantage of that. All right, so again, here, if I go to this view instead, don't need that, do need that. So again, your student view of the Mastering A&P is going to be slightly different. This is the instructor view, but it should still have your introductory assignment, which hopefully you've done and all of that kind of stuff. But over here, when you go to study area and you launch it in its own pop-up window, this is where you find the interactive physiology. And again, if you notice, look at all of the directed tutorials, all just for the cardiovascular system. Cardiac output, cardiac cycle, right? Intrinsic conduction system. There's a great resource. I strongly encourage you to take advantage of this. And that's why I've given you that worksheet for the first one to do, because I thought that that would be a great incentive to force you to come on here, look at this, see this is here, say, ah, this is really cool, and this really helps me to understand the material, and that will be awesome. The other thing, oops, wrong button. If we go back here, is remember this is also where you will access the PhysioX. The PhysioX exercises are located here. So again, both exercises 5 and 6 involve the cardiovascular system, but remember we are doing 6 first with 5 different activities. Then you're doing 5 that has 7 different activities. And at the very end, you're going to come all the way down here to blood analysis and do blood typing. For the last one, you just have to do activity 4. Again, you're welcome if you're interested in blood cholesterol or hemoglobin determination or any of the rest of these things, you're welcome to do that. But this is the one that you are turning in from me. So you'll do a lab report for this, 1 lab report for this one. Activity 6 has 5. And lab reports and the cardiovascular has 7. And remember, you don't have to print those out. Just save them as PDFs when you're done with them. And those are the PDFs that you'll drop off in the Dropbox. All right. So I got several questions on that. So I want to make sure we understand and are comfortable with that. Any questions on those lab simulators? Because you got a lot of them during this section. So any questions on that before we dive back? Excellent. Stunned silence. That's what I love. My microphone is on. You guys can't hear me, right? You would tell me if I wasn't, you couldn't hear me 15 minutes in?
Oh, yeah, I hear you.
Perfect. Excellent. All right. Let's talk game plan then. Today, in the last class, we mostly talked about the anatomy of the heart. So now we get to talk a little bit more about the physiology and the function of it and how that's going to work. That'll also give us a chance to talk more about the cardiovascular anatomy, which is going to be important for us to make sure that we understand that. And we'll talk about some of the tools that you have available for you for that. Although I think we went over them, I want to go over some more. A bunch of assignments coming up over the next couple of weeks. Your first, if you haven't gotten your lab manual yet, your first lab assignment is due on Tuesday. As a quick note, let me try that again. The bookstore has the lab manuals in stock. I have spoken to several students who say they have not received their email from the bookstore yet saying that it is ready for pickup. But I've also had several students tell me that when they call the bookstore has it and they were able to go and get it. I know the email says, don't call the bookstore. I'm telling you, call the bookstore. Be the squeaky wheel, get your cheese. All right, so do that and get the lab manual. The first unit assignment from that is due on September 1st. Your first a lab simulator, the cardiovascular function from the Labster is due. Remember, for the Labster activities, you have to do the entire activity, and for full credit, you have to get it at least 80% correct. So if you don't, remember, it's a learning activity. You have the chance to do it many, many times. Right? Share it with your loved ones and kids. Notice I didn't put the kids in the loved one category. You get the idea. And again, do those that way. Also, your student info sheets, if you haven't filled those out, those are done as well. The interactive physiology and PhysioX, your other two lab simulators, are due on Thursday. The following one, we have our Unit 18 review due, our PhysioX exercises are due, and our cardiovascular exercise. That's those driving directions that we talked about with that blood vessel. We went over that at the end of the last class. No, great question. The PhysioXs, again, I'm looking for completeness. I'm looking for time and effort into them. I'm looking for time and effort, and as long as you're doing time and effort, it's a learning activity. It's okay to get something wrong. Same thing with the Labsters. That's why I set it at 80%. As long as you get 80%, if you're paying any attention, you should be able to get 80%. It shouldn't be too hard to get 80%, unless you're really unclear on the concepts, in which case, then study it, because that's the point of this. All of these things are really just directed studying, right? All of these hoops that we're making you jump through are really just ways of directing your studying to make sure you get this information. The Master NP introduction should be set up to auto sync, but sometimes it takes some time. I actually haven't checked it yet to make sure it's auto sync set up, but if you did the Master NP introduction, I'm sure it is there, and I will check that. That's one of the things that I have to check this weekend too, so I will do that. Map intros. Add to my to-do list for this weekend. All right, excellent. Yes, as long as you get 80% correct, you will get the full 20 points for it. All right. All right, excellent. So again, those are the activities that are going on, another PhysioX, and then on September 15th, we have our first lab and lecture exam. So it is gonna go quickly. All right. Oh, that's the last thing I wanted to show you. Hold on. We'll do that with this one here. All right, we're done with that. Let's go, actually, hold on. Let's bring that back. Let's do that. I am in the process of grading your proctorio quizzes. A lot of you waited till yesterday to get them, and I hadn't had a chance yesterday. It was a very busy day, so I have not had a chance to grade them all. I'm still in the process of them. But there are some notes that I wanted to say. First and foremost, when you take these types of quizzes and stuff, I often put comments in them. When I put those comments in them, you should see a couple of things. When you're on your home page, you should see a blue dot next to your grades. When you click on your grades and go into your grades, if I put a comment there, you should see that there is a comment right here, and you can actually click on it and see what that comment is. The other place you should see it when you are on your home page. On your home page for the Canvas site, notice there's the feedback that we have here when things are graded, and if I put comments or things like that on there, then those are there. Make sure you're checking those things. I've been looking at the proctorio, and if there's been some issues with that, I've been mentioning it. Nobody's losing points on this proctorio quiz, because that's the point of it, to see how it works. But there are some notes and things that I wanted to talk about. Again, I realized after talking to my 430 class yesterday that yes, those videos that I've showed of people scanning their areas, that looks really fast. But that's, remember, because I just recorded the views that I got from them, and it's sped up. So you need to slowly scan your area. And again, I don't care, right? I don't care about being up here and seeing everything that's in your room. I want to see what's on your workspace. So make sure you are showing me your whole workspace and everything that goes around in your immediate area for that. That is what I'm looking for. And again, if you don't do that, that's 10% right off the top of your grade. Make sure you have enough lighting so that I can see you. And again, sometimes that can be tricky because if you're in a really well-lit room, but you have a really bright window behind you, then often being backlit, your face is going to be very dark. Remember, you have a camera picture up there. And I know it's awkward to be thinking about yourself being recorded, but make sure you can see yourself clearly on there, because if you can't see yourself clearly on there, that means I can't see you clearly either. And not only is that a lighting issue, but it's also a location issue. If all I'm seeing is your furrowed, forehead as you're working on this, then I can't tell what you're looking at and what you're doing. You need to make sure that you stay as focused in the center of this as possible. Oksana, you had a question?
Yes.
Yeah.
If I got 8.5 out of 10 for the proctorio, it means I didn't do it properly, or it's about the questions?
I did not take any points away from anybody for the way that they did the activities, whether they didn't scan their ID properly or didn't scan their work properly. If you lost points, it was because of questions you got wrong on the exam. So nobody lost points for the process. It was just questions wrong on the exam. Okay?
Okay, thank you.
Yep, excellent. And like I said, I'll get those posted shortly. Head centered in the camera, headphones, no earphones. Again, on a real exam, you cannot wear earphones during the exam. You can't have your AirPods in or anything else on. I know especially some of us in our environments, we like our noise canceling earphones so that it allows us to concentrate better, but there's no way for me to know what's being pumped into your brain right there with that. So you cannot wear any earphones during that. And again, I think we talked about it in this class. Again, it's hard when I go back to back. Make sure when you're showing your ID, you are showing the ID to the camera, right? Not holding it up to the screen. Just hold it up to the camera, line it up with that green square, and then that's what you need to do for your ID for that. All right? Any questions on that process? Like I said, as soon as I finish getting them graded, I will post those with the comments, and if you have any further questions after that, please feel free to ask me. All right, the other thing that I did actually, and again, some of you have still not filled it out, the student info sheets, or I still haven't had a chance to go through them, but I have gone through most of them. So there is some statements and comments questions that I wanted to address related to that. I was very impressed with you guys in general as a class. Normally there is a predominant amount of pre-nursing students, but actually you guys, while of course, as always, there's a large amount of students, there's also a wide range of other interests in this class. So that's really fun. We got everything from phlebotomy to radiology and pretty much everything in between, which is kind of cool that way. A lot of you are just interested in learning everything about everything, which again, as an instructor, you love. But of course, a lot of you, a couple people, the endocrine system, which I thought was really exciting. Because again, that tends to be one of the more challenging sections of this, but a good, again, sprinkling of different topics there. One of the questions that I got asked that I thought was really important is, what do you need from 430 to move forward? And what I would say is two things. The first thing that I would say is your study skills. As far as I'm concerned, 431 is really just a continuation of 430. The same way I ask questions, the same way I tackle the material, the same way I present the material, the same way I do the quizzes and the exams and everything else in 431 is exactly the same way that I did it in 430. Now, if you had me for 430, that's an advantage because you already know what to expect. But if you didn't, that's okay as well. The goal of all 430 instructors, and myself included, is we don't pick the stuff randomly out of a hat that we are holding you responsible for in 430. We taught you the things that you needed in 430, but also the things that you're going to need going forward, right? Yes, in this class, I am not going to ask you for a terrible number of bones and bone features. Right? But when we talk about the endocrine system and I tell you that the pituitary gland is located in the hypophyseal fossa or within the sella turcica, anybody remember what bone that was a bone feature of? The sella turcica is a bone feature of what bone? Sphenoid. There you go. The sphenoid bone. Excellent. Right? So there is going to be some callback to things like that. We talked about, some of the holes like the jugular foramen, the carotid canal. Obviously, those are going to be passageway for blood vessels we're going to be talking about. Right? I'm not going to be asking you for muscle attachment points, for this, but are the muscles that are used for respiration going to be important? Absolutely. And probably the most important thing you hopefully got from 430 was the autonomic nervous system. Right? Your autonomic nervous system is what controls your smooth muscle, your cardiac muscle, and your glands. Right? Cardiac system, respiratory system, digestive system, urinary system. All of those things are things that are controlled by the autonomic nervous system. So knowing the anatomical and physiological functions of the sympathetic and parasympathetic nervous system is going to be important. And we're going to talk about it a lot. So making sure that you understand the pathway by which the sympathetic nervous system would increase the heart rate and how it would do it and what neurotransmitter it's going to release are things that you should have learned in 430 and we will be using in here. Now, the good news is all this information is in your book. So if you didn't get the autonomic nervous system as well, if your teacher didn't hit the histology as hard, and again, I've fully admitted to a couple students who have come to the 5 for 5, right? I don't know how you felt about Tuesday's lecture, but I hit the histology on the heart pretty hard with all of our magical terms and things like that, and I totally do that on purpose. Right? I know some people are going to know, especially coming in the fall, because some people just finished summer just 2 weeks ago, and so that information was still pretty fresh. But it's a way of either reminding you or pointing out to you that microscope work, understanding tissues, understanding histology is something that I think is very important and an important part of anatomy and physiology. So if those were things that weren't emphasized in your 430 class, then the good news is you have time to go back and look at those things and learn them. But we're going to continue to talk about those things again and again and again in this class. So histology is going to be a huge part of this system and the digestive system. And oh my God, the next system when we're doing the endocrine system and we're looking at all the endocrine glands histologically, it's going to be brutal. So if you didn't get that, now's the time to start working out. That's why that histology atlas that we talked about is so important, so that you can look at a good example of what the adrenal gland looks like. So you can see the zona glomerulosa and the zona reticularis and the zona fasciculata and be able to distinguish them from each other as well as from the medulla of the adrenal gland, so that you can understand the classes of hormones that are produced there and the specific hormones that are produced there and the targets of those and how they're regulated and what their specific functions are and all those things we're going to get to do in the endocrine system. So these things are definitely important. At least I didn't pull the things that I taught you in 430 randomly out of a hat. And that's also why me and most instructors have a cumulative final exam, because you need 430 to move forward into 431. All right. I know several of you are taking microbiology concurrently. And I have had students in the past that have done it and been successful. In the classroom, I've also had students who have tried it and not been successful with it. So the one thing that I want to remind you, actually, there's two things I want to remind you about for those of you who are doing this. Look, we do not, what's the term I'm looking for? We do not stop you from doing this, right? It's not illegal. We don't, you are capable of signing up for these classes, and this is college and you're adults. But here are the two things I will remind you. You are in this class because you were successful in 430. And the reason you were successful in 430 is because you put a tremendous amount of time and effort into it. You know how much work this class takes. And microbiology requires just as much time and effort, especially because I'm fairly certain about 2/3 of the stuff you guys talk in microbiology is completely made up. Prions, right? These unbreakable proteins, I mean, that's totally made up, right? But these things take a lot of time, and it takes time to be successful. And the two things I will remind you is that for most of you, you're taking it because you're rushing towards some goal that you have. But remember, for most of you, grades matter. 2 A's in 431 and micro is much more important than an A and a B is going to be for you. So it doesn't help you to rush through these if you don't get the grade that you want. All right. The other thing that I will remind you is not only is it twice as much time as it is to just take 430 by itself, but the other challenge you're going to have, and this first section is a perfect example of this. Today is the second day of class, and one of the things that we would have done at the end of class today was done our heart dissection, right? Tuesday, when we came back, we would normally do an ECG activity where you get to put the electrodes on each other and you get to measure an ECG and then measure the timing of them and then have the person run around the classroom and then do the same thing again. We get all this physiology and we do it in the classroom. I've done the best I can to recreate that the best I can with the lab simulators, but notice we're not taking class time to do those activities. So those are activities that you're now doing outside of the class time to work on those things. Yes, like last class, we may be ending a little bit early in some classes because those are normally days that we had a lot of physiology, so usually I don't lecture as much on those days, but it's not going to be enough time to complete those activities. So completing this class successfully is going to typically require even more time than it normally would because of all the outside hoops you're going to have to jump through to try to maintain the integrity of this class by having things like these lab simulators. So like I said, by no means is it not possible to be successful in both this and microbiology. I just want you guys to be fully aware of what you are getting yourselves into on this so that you can make that decision before you get too far into it. Remember, you only get to take these classes 3 times, right? And so if you get halfway through the class and realize, oh, I was wrong, I can't do both, so I'll drop microbiology and stay in 431 because Professor Slutsky is awesome. Then the problem is if you've been really struggling in 431 because of that, you may not have enough time to make up the grade to get the grade that you want. So I just want to make sure you guys are aware of what you're putting yourself into. Make sure you understand the time commitment that you have involved in this so that you can be successful. Again, if you're hunkered down in your house, getting meals delivered to you, right, never leaving, and you have nothing but time on your hands, can you be successful at these? Yeah, possibly. But just be aware of the time commitment and the other things that you have going on in your life because I want you to be successful. I don't want you to waste your time here. All right? Questions on that? All right. One person asked if it was possible to sneak on campus and be able to see the materials. I would love to have the opportunity for you guys to be able to do that, for us to be able to do that. It's just not something that's possible right now. Who knows? Maybe after Thanksgiving, maybe things will change. Maybe by the end of the semester, we may have a chance to do that. But as I've told several students, there is already rumblings on campus that they're planning on going fully online for the spring as well. So I think if you ask me what the 2 choices were, do you think it's more likely that we'll be on campus by the end of this semester or we'll be online next semester? I would say right now it's leaning more online next semester. So again, I think that we may be here for a while. However, while we can't sneak on campus, remember you do have that open lab. Jeff does have a lot of resources, a lot of the materials, a lot of models. He's got a couple cameras, he's got a couple things set up, and he is able to share those things. Yes, so if you go to the Friday Open Labs for 1 hour of Open Lab, we'll get you 1 point of extra credit up to 15. But remember, as I also said, 15 hours in Open Lab, if you're using your time wisely there, is going to get you far more than 15 points on the exam. So yes, it gives you, take that carrot of the 15 points. But if you spend those 15 hours in Open Lab studying it, you're going to do way, way better on the exam. All righty. Last and also least, my information, I have been teaching, this is my 14th year at American River College. However, between other Los Rios schools, and I spent some time teaching at Davis and some Woodland, I was at Yuba for a bit. I have been teaching for about 20 years now. I did both my undergraduate and graduate work at UC Davis, and my PhD is in the field of neuroscience. In particular, I studied the development of the visual system. If you do the 5, yes, so yes, it's not all or nothing for the Open Lab. If you get to 7.5 hours, you'll get 7.5 points. So it's not just all or nothing. If you only get to 3 hours, you'll get 3 points. Absolutely. All right. Any questions on any of that? All right, excellent. I promise we will not continue to take half an hour for introducing and introducing the class. But again, for this first week, as we're learning this online and getting to know each other, I think it's worthwhile because, again, I want to make sure everybody's successful as we move forward. All righty. With that in our pocket, let's pick up where we left off with the lecture. When we left off in the lecture, as we were talking about how the heart anatomically is really these 2 side-by-side independent pumps involved in moving blood, first the oxygen poor blood and the oxygen rich blood, to different parts of the body. Now, of course, with 2 pumps, you would think that we would therefore have 2 circulations. However, in fact, there are actually 3 circulations of the body. What are they? Someone give me an example of one of them. What is one of the 3 circulations of the body?
Lymphatic system.
I'm sorry?
Lymphatic.
Oh, excellent. You are absolutely correct. The lymphatic system definitely is one of the systems involved with circulating. And I guess when I'm talking about here is really our blood circulations of the body. So you're right. Lymphatic is one, and we will talk about that. There you go. There's one of them. Pulmonary. Let's start with that one first. Pulmonary circulation is one of our 3 circulations. Excellent. What is the pulmonary circulation? From what to what to what? From the heart to where? To the lung. There we go. Excellent. To the lungs. And then, of course, back to the heart. Excellent. And, of course, what is the function of the pulmonary circulation?
Oxygen. So that
Give gas.
Yeah, it's to, exactly. Take blood to the lungs for gas exchange. Right. Excellent. Move that there. Oh, no. Move that there. Move that there. Perfect. Excellent. Excellent. For gas exchange. Perfect. I saw, what's another one? Systemic. I see systemic. Circulation. Excellent. And what's that one? From the heart to where?
Body.
To the body, absolutely. And then back to the heart. Excellent. And what is its function?
To give oxygen to organs and muscles and everything.
Right. Excellent. I like that. Let's simplify that. Good stuff to the cells of the body. And take the bad stuff away. Excellent. All right, so we take the good stuff to the cells, bring the bad stuff away from that, right? Clean that bad stuff on the way, going to the kidneys and things along those lines. Excellent. So if we got into the lungs, we've gone to the body. What does that leave us with? What could our 3rd circulation possibly be?
Cardiac.
Yeah, kidney is a good guess. Nervous system is a good guess, right? Our brain is big, important, but it turns out the kidney and the brain are just part of the systemic circulation. So really, as someone mentioned, it is the heart or our coronary circulation, cardiac, or we can say coronary circulation. All right, so that goes from the heart to what?
The heart.
Yeah, to the heart. And then, of course, back to the heart. Well, that doesn't seem to make a lot of sense. But really, in this case here, when we're talking about the heart, we're really talking primarily about the muscular wall of the heart. And then back to the heart chambers, right? We're really talking about from heart chamber to the muscular wall of the heart and back to the heart again. And of course, what is the function of this coronary circulation?
Keep the heart alive.
Say again?
It's hard.
Keep the heart alive.
Yeah, absolutely. I like that. Keep the heart alive. Right. Again, kind of like the systemic circulation, good stuff too, and bad stuff away. However, even more than that, it's really about getting oxygen to and CO2 away. Because as we know, our cardiac muscle is highly, highly dependent on oxygen. And if it is deprived of oxygen for even a brief period of time, it can be damaged. But that's no big deal, right? Because we can just repair all of those damaged cells in our heart when we need to, correct?
Nope.
No, as we talked about, that cardiac muscle does not regenerate. So maintaining oxygen-rich blood flow to our heart is vitally important. In fact, as that oxygen-rich blood leaves our body in the systemic system, It is the very first thing that it feeds blood to is to the heart. Excellent. So now we've talked about some functionality, but let's go back on this. Oh, I'm not gonna, oh, I am good. Excellent. And take this one step forward. What are the blood vessels involved in our pulmonary circulation? Of the great vessels we've talked about, the blood that, and again, we can start with from the heart to the lungs and back to the heart. So when it leaves the heart, what chamber of the heart does it leave?
Right ventricle.
Excellent. From the right ventricle. And then it goes into what blood vessel? What's the great vessel that receives the blood?
Pulmonary trunk.
Pulmonary trunk.
Pulmonary.
Excellent. All right, and then from there it goes to the lungs. What is the terminal terminal great vessel that receives, or vessels that receive the blood coming back to the heart? Pulmonary veins. Excellent. And remind me again how many there are?
4.
Excellent. 4 pulmonary veins. And of course, they feed into what chamber?
Left atrium.
Left atrium. Excellent. So that right there, right, with the intermediate blood vessels in between, the pulmonary arteries and arterioles and all of that stuff, and then coming back out, is our pulmonary circulation. What are the blood vessels involved in our systemic circulation? Again, from heart to body, back to the heart. Where does it leave the heart?
The ascending aorta. Oh, left ventricle.
There you go, left ventricle. And then you're right into the ascending aorta. Oops. Aorta, excellent. And then again, all those elaborate branching blood vessels to the body. Then it comes back to the body via what blood vessels?
Veins.
True, what vein? Yeah, the veins bring it back to the body and bring it back to the heart. And what great vessels receive that blood from the body?
Inferior, superior.
Excellent. And I'm going to abbreviate this here because we've written them a couple times. Superior vena cava and the inferior vena cava. Excellent. And they feed that blood into what chamber?
Right atrium.
Right.
Atrium.
Right atrium.
Right, atrium. Excellent.
Atrium. Right
Perfect.
atrium.
Not a tremendous amount of new information there. But let's talk about our cardiac or coronary circulation. As I mentioned, the heart is the very first structure that receives blood as that oxygen-rich blood leaves the heart. And what is the great vessel that receives that oxygen-rich blood again? When
Pulmonary.
oxygen-rich blood leaves the heart, what blood vessel receives it?
Aorta.
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 So the ascending
Aorta.
aorta. As I mentioned, the ascending, well, I didn't mention this, but as I mentioned, the heart is the very first thing that receives oxygen-rich blood as it leaves the heart. There are only 2 blood vessels that come off of the ascending aorta. Anybody happen to know what those blood vessels are?
Coronary artery.
Anyone know what the blood vessels are that come off of the ascending aorta?
Is it common carotid?
Nope. Common carotid comes off, the common carotid, brachiocephalic, I should say the left common carotid, the left subclavian, the brachiocephalic come off of the aortic arch. There are only 2 blood vessels that come off of the ascending aorta. Let's cheat.
Left and right coronary arteries.
There you go. Excellent. They are indeed the left and right. Say it again.
Coronary artery.
Coronary arteries.
Arteries.
Excellent. We'll look at the pretty picture of this in just a second. The left and the right coronary arteries. These left and right coronary arteries are what feed the muscle of the heart. Excellent. All right. Now, as that blood is fed into the heart, it then has to leave the heart muscle, and there's a big, large, loose vein. I don't think we talked about this in the last class, but remember we said a trunk is a large artery that is short and branches almost immediately. Well, a sinus is another type of special blood vessel. A sinus is a big, huge, loose vein that allows it to accumulate a lot of blood. So what is the special blood vessel that all of the blood from the muscles of the walls of the heart are fed into? There you go, Lisa's got it. It is the coronary sinus. And that coronary sinus, once it receives the blood, and I guess we should go back a step, it is the left ventricle that feeds into the coronary sinus. A left ventricle that feeds into the ascending aorta. And that coronary sinus, any idea what that coronary sinus feeds its blood into?
Right atrium.
Right atrium, absolutely.
Atrium, sorry.
Right atrium. Remember how in the last class I told you there are 3 blood vessels that feed into the right atrium? What are the 3 blood vessels that feed into the right atrium?
The superior and inferior vena cava, and then
what's the third?
No.
Coronary sinus. Excellent. Coronary sinus. There you go. Excellent. Like I said, let's cheat and go back and look at this. See if we can get a good picture. I know I have a good picture coming up, but let's, oh, this was on screen. I didn't realize that. Okay, cool. All right, here, let's do this. Put that down, put that down, do that. All right, everybody got this? Quiet reflection, so excellent, that means yes. So I can clear that. That I had done that on the whiteboard, sorry. Excellent, so let's go back to one of these pictures. Not back far enough. This is what I want. Perfect. Excellent. So here we have the heart. Again, here we have the right atrium. Here we have the left atrium, these little dog-eared looking things, right? That nice auricular surface. Here we have the ascending aorta. Coming off of the ascending aorta, you need to know all 3. You need to know all 3 blood vessels that feed into the atrium. Coming off the heart, you have the right coronary artery that comes off this way, and then the left coronary artery actually is very shy. It sneaks behind the pulmonary trunk right to here, and as soon as it pokes its head out, it branches and goes into those 3 directions there. So like I said, these 2 are the only 2 blood vessels that come off the ascending aorta. All right? No, superior and inferior vena cava. There's no right and left, superior and inferior. All right, if we open up the heart, notice now they've done it. Well, actually, let's, I think that other picture I like better. So many pictures we went through. Let me see if I can find it. No, that one doesn't do it. I guess that is the best one. So what this hole here is supposed to represent is that coronary sinus coming off the back. But I have a better picture. I'll show it to you in just a minute. So let's do that. All right. Bing, bing, bang, bang, bum, 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 bum. There we go. All right, let's start here. Let's take a look at our 2, 3 circulations. Here are the first 2, systemic and pulmonary. And remember, one of the things that we talked about is there are far more systemic blood vessels. Most blood vessels in the body are systemic. And as such, as we know, the arteries are going to be oxygen rich, and the veins are going to be oxygen poor. However, the reason we can't use the oxygen capacity as the characteristic for determining arteries and veins is that pesky pulmonary, because it's the pulmonary that are backwards. All right, where the arteries are oxygen poor, because they're going to the lungs to get the oxygen, and the veins are oxygen rich. And they do it that way. But here we see the circulation, the capillaries where they turn around, and all the pretty stuff that we kind of had already talked about in the last one. So there's not too much new stuff here. Another pretty picture showing and representing it as well. And here we get a better sense of which chambers are involved. So again, our pulmonary circulation starts in the right ventricle, out to the lungs, back to the left atrium. The systemic circulation goes from the left ventricle out to the body and back to the right atrium. But here we get a better sense of our coronary. Notice here, they've removed that pesky pulmonary trunk. So here we can get a much better view of those only two blood vessels that come off the ascending aorta, the right coronary artery and the left coronary artery. And notice the heart is one of those areas. Remember when we were talking about the arms, we talked about how there was symmetry in the arteries and the veins. So the brachial artery is right next to the brachial vein. The arms are completely symmetrical as well. The blood vessel structure in your right arm is identical to the blood vessel structure of your left arm. But the same is not true for the heart, which if you think about it makes sense. We already saw that the heart is asymmetrical in that the left side is thicker, the left ventricle has a different orientation than the right ventricle. So there are some asymmetries. So it isn't surprising that we see these differences. Notice as we work our way through our coronary circulation, the right coronary artery comes out and notice basically goes in that indentation. If only we had a fancy term for an indentation. Any idea? Is there some term we learned for a nook, a type of indentation, maybe something associated with the nervous system that we used?
Sulcus or gyrus.
Sulcus, absolutely. Sulcus is that indentation. Notice it sits right here in the atrioventricular sulcus and runs basically all the way around to the posterior side of the heart. Whereas, as I mentioned, the left coronary artery is much, much shorter. Basically just poking out past the pulmonary trunk, and then it branches. When it branches, the first branch goes down the indentation along the front of the heart, in between the ventricles. And any idea what we might call that indentation between the ventricles on the front side of the heart?
Fossa?
Okay, fossa is a good one, but remember we just learned sulcus, so fossa probably wouldn't be bad, but sulcus is probably better.
Longitudinal.
All right, well, it's on the front between the ventricles and it's an indentation. How about anterior interventricular sulcus? And guess what we call the artery that sits in the anterior interventricular sulcus?
Anterior intervertebral artery.
Artery, there you go, excellent. And guess what we call the vein that sits in the anterior interventricular sulcus?
Same name.
Anterior interventricular vein?
Interventricular vein.
That would make so much sense, but what do we know about anatomists?
Hates us.
They hate us, absolutely. So while we have an anterior interventricular sulcus and an anterior interventricular artery, do we have an anterior interventricular vein?
No.
No. What do they call the vein?
Oh, it's a middle cardiac?
Nope.
No, it's a greater coronary vein.
Close. It's the great cardiac vein.
Cardiac vein.
Yeah.
Oh,
so, yeah.
great.
So again, anatomists hate us. And this is a perfect example of that. We have a sulcus, we have an artery with the exact same name, And they could have done the exact same thing with the veins, but they didn't because they hate us. Excellent. Notice also there is another branch, and we'll write these out. Again, it's again good flow chart type stuff. Notice again that left coronary, switch back to my highlighter, left coronary artery comes out and then branches, and in one branch becomes the anterior interventricular artery. The other goes in that sulcus between the atria and the ventricle around the circumference of the heart, and so it is called the circumflex artery. All right. We'll look at the heart and talk about the blood vessels of those in a little bit more detail later. But one of the points I want to emphasize is that the heart is only about 1/200th of your body weight. So if your goal is to lose weight, ripping your heart out of your chest isn't a good way to do that. You're only going to lose, 1 pound, less than 1 pound from doing that. However, as small as it is, as I mentioned, it is the very first structure that receives blood, and it receives about 1/20th of your blood supply. 10 times more blood than it should get based on weight, simply because it is so vitally important to keep that oxygen flowing to the blood, oxygen flowing to the muscle, because as we said, if that cardiac muscle is deprived of oxygen, even for a brief period of time, it could be severely damaged and it doesn't regenerate. Like I said, we'll talk about the blood vessels more, but notice all the blood from all the veins drain into this big, huge sinus on the back, and that sinus feeds into the right atrium. Like I said, I have some better pictures we'll look at, and we'll talk about the anatomy of the heart. It's one of the areas where we have asymmetries from the left and the right, asymmetries in the arteries and the vein, and it's a pretty important structure. Like I said, arms, legs, it's pretty symmetrical. There are a couple wonky things, but mostly symmetrical. Anyone know the other big place where there's dramatic differences between the arteries and the veins? There's really two big areas where there's big differences between the arteries and the veins. One is the abdominal pelvic cavity, and we'll talk about why that is in a little bit. Anyone know where the other area is? In the skull, in the head, huge asymmetries. Yes, the heart receives about 1/20th of your blood supply. That is correct. While you're sitting at rest. All right, excellent. All right, we talked about this a little bit in the last class, but let's do it a little bit more here. Our anatomy of our contractile cardiac muscle cells. Our contractile cardiac muscle cells are very much like skeletal muscle. All right, they're striated. Remind me again, what causes the striations in muscle tissue? What is the organelle? Let's start easy. What is the organelle that produces the stripes of a muscle cell? What's the organelle? That's not an organelle, Ryan. You got the right idea. And actually, that was the first question I was asking for, but I took us back a level. When you put a bunch of sarcomeres together, you get an organelle. What organelle was that again?
I forgot.
Anyone remember? I'm trying to cheat by looking at the picture to see if it's there, but it's not actually there. Interesting. Even
it's
better.
not here.
Yes, summer was a blur, which is one of the reasons why I said not to take summer school, which I did many, many times. Excellent. Somebody else. There it is, myofibril.
Oh, finally.
Myofibril is the organelle. Excellent. But Ryan is absolutely correct in that the organizing structures of the myofibril are the sarcomeres. All right, so we have the sarcomeres, which are those organizing structures made up of lots of proteins. And what was the generic term we used for all of the proteins that made up a sarcomere? Myofibrils, excellent. Although I guess someone said that. I'm not sorry, not myofibrils. Myofilaments are the proteins. And remember, there are three types of proteins. Contractile proteins. Let's start easy. What were the contractile proteins of the sarcomere again? Myosin and actin. Excellent, excellent. Which one was the one that had the heads that had the motors in them?
Myosin.
Myosin, excellent. Remind me again what a motor is. What is a motor? Again, I have to start coming up with some different examples because normally what I say at this point is most of you got here today to school today because of a motor. And when your car stopped finally in the parking lot, one of the things you did to thank it was to get out and give your hood a big hug, including laying your face right down on the hood, right? Giving it a nice big smooch to thank your car for getting you there. How many people kiss the hood of their car when they get to their destination? Anyone? Why wouldn't you want to do that? Every time, Ryan. Excellent. Everyone but Ryan. Why don't you want to do that? What's wrong with the hood of your car when you get to your destination? There you go. It's hot. Absolutely. Remember, a job of a motor is to convert chemical energy into mechanical energy. Right? With our myosin head, the chemical energy is the ATP. The mechanical energy is that power stroke, that pivot towards the M line that it takes, that shortens the sarcomere and generates the force, produces the tension that we need, right? Everything that goes along with that. It does that. And like most motors, like the motor in your car, it isn't 100% efficient. Much of the energy from it is lost in the form of heat. In fact, some muscle cells, those myosin heads, only have a 25% efficiency. That means 75% of the energy is lost in the form of heat. All right, so we have those myosin heads grabbing and pulling, shortening that sarcomere, providing that tension. We had some regulatory proteins that affected when the muscle contracted and when it didn't. What were our regulatory proteins again? Troponin and tropomyosin, excellent. And we had some structural proteins. What were some of the structural proteins again?
Z-disc.
Myosin forms the M line. Titin, that gives the elasticity to it. And what was the other one? Z-disc, excellent. Right? All of these were examples of myofilaments that are precisely arranged into sarcomeres. And those sarcomeres form a repetitive pattern that is the myofibril, and that is the organelle. That is what allowed our skeletal muscle to contract. And the good news is our cardiac muscle has these as well. So our contractile cardiac muscle contracts in a very similar way to skeletal muscle, which is a good thing because we learned how skeletal muscle contract, so we're going to understand how cardiac muscle contracts. Of course, what's the key word in that sentence?
Very similar, not exactly.
Similar. Similar means not exactly. There are going to be some subtle differences on that. Absolutely. But in general, it's going to work in a very similar fashion. Not identical, and we will see some of those differences, and we will talk about that. But there's some other ways that it's not like skeletal. It's shaped. They're shorter cells. They are branched cells, right? As we see here, and here this picture does a really nice job of showing this. Notice here in the space in between, we see those connective tissue. That nucleus is actually the nucleus of the connective tissue, right? And this is that fibrous skeleton that we saw inside of here. Notice we can see the striations, but there's also this really dark stripe that runs parallel to the striation. Anybody remember what that thing is called?
Intercalated discs.
Excellent. It's the intercalated disc. That intercalated disc is what strongly holds those branching cells together. And here we see that nicely on the illustration here. As we talked about in 430, or you can cheat and look at the pretty picture from the textbook here, there are a large amount of cell junctions in our intercalated discs. Primarily, what two types?
Gap and desmosomes.
There you go, the gap junctions. And what did the gap junctions do again?
Electric signals.
Right, they're fluid-filled pores allowing the cells to basically be wired together. And you're absolutely correct. They're basically forming an electrical synapse. These cells are hardwired together so that what happens to one happens to the other, so that when our heart pumps, it works as one single unit. Right? In my arm, if I want a muscle cell to contract, I have to send a signal to it and tell each individual muscle cell to contract. That is not the same thing in the heart. I produce one signal and it can spread through these gap junctions to all of the cells. What's the second type? I know you said it, but remind me again. The second type of cell junction you find here in the intercalated discs?
Desmosomes.
Desmosomes. Remember, the desmosomes are the ones that form that tight spot welding type of connection that is going to hold these cells together from the massive force that it is generating as it pumps the blood. No, we do not have graded recruitment the same way. We don't have motor units. No motor units in our cardiac muscle, so we don't have to worry about recruitment the same way we do in skeletal muscle. Great question, but no motor units, so no recruitment. Nope, so again, that's one of the ways that it is different, absolutely. Excellent. Again, it is involuntary, can't be voluntarily controlled, right? But like I said, not identical. Notice we do have transverse tubules. We do have sarcoplasmic reticulum, but we don't have the enlarged terminal cisternae. We don't have triads that form within this region as well. And one of the implications of this is, remember, those terminal cisternae allowed us to release a tremendous amount of calcium into the cell from that sarcoplasmic reticulum, that jailbreak of calcium. And as we know, calcium makes cells do wonky things. Well, here in this case, we are going, without those triads, our cardiac muscle has to rely more on extracellular calcium. Whereas almost all the calcium for our muscle contraction in a skeletal muscle comes from inside the cell, our cardiac muscle has to rely a lot more on calcium from outside of the cell. All right, excellent. And again, much more reliant on oxygen to produce ATP, so not surprisingly, it has much more mitochondria in it than our skeletal muscle cell as well. All right, excellent. That is a good point, a good starting point. We, like I said, know some things about skeletal muscle and skeletal muscle action potentials and skeletal muscle contractions. So we are going to use what we know about that to relate the similarities and the differences between our cardiac muscle and our skeletal muscle. Things get a little fun from here, and it's been an hour, so this is a good break time. Let's go ahead and take our first break here. We will come back at 1:20, so we will restart at 1:20, and I will start the recording at that part. If none of the things we've said over the past 5 minutes sound familiar or you remember, I would strongly encourage you to grab your textbook and take a quick peek at at skeletal muscle, so that as we talk about skeletal muscle in the review part of the things that we already know about it, we can then focus more on the cardiac muscle. All right? Questions on that? All right, I will see you in 15 minutes then. Any questions before we dive back in? All right, excellent. Stunned silence means I completely understand everything. Please move forward, and also please make the test harder. Excellent. So I like that's where we're at. Perfect. Excellent.
No, don't make it harder.
So let's talk about how cardiac muscle and skeletal muscle contract and how they are different from each other. As we kind of already talked about, because our cardiac muscle cells have those gap junctions, they're all connected as one unit. So there are no motor units connected to different parts of it. It all basically is going to work together as one. There's also some big differences in lengths of contraction. If you remember when we talked about a contraction of a skeletal muscle cell, that entire contraction process, remember something we referred to as a twitch, which is hopefully something you remember. If not, I will strongly remind you of that. So I spell it correctly. From latent period to the end took about 20 to 100 milliseconds. However, the contraction length from a single stimulation of our cardiac muscle cell, that contraction lasts over 200 milliseconds. Why might there be a big difference in the amount of time for the contraction of these muscle cells? Why would you want cardiac muscle cell to have such a longer contraction time?
So make sure you get all the blood out of the heart.
Yeah, it makes it a much more efficient pump. How many people here have blown up an air mattress before? Anyone? One person, a couple people nodding, absolutely. And again, I don't mean the way you do it nowadays where you plug it into the wall and you push the button and it fills up. No, I mean old-fashioned hand pump, right? When you're using one of those old-fashioned hand pumps, is it more efficient to make a whole bunch of really small, really short, really fast pumps of that air pump? Or is it more efficient if you're taking a full, complete cycle with that pump? That longer sustained contraction is much more efficient for the pumping motion. So yes, by having a longer contraction, as Nick said, it makes it for a much more efficient pumping motion to move that blood out. Excellent. Remind me again what the absolute refractory period was.
It's where you can't, action potential is like, it can't depolarize.
Yeah, absolutely. Right. Remember, one of the things we talked about with skeletal muscle, and we talked about this with nervous tissue as well, it is irritable. It responds to a stimulus. It receives a stimulus and responds by producing an action potential. But remember also we talked about when an action potential is produced, there is a brief period of time where it loses that excitability, it loses that irritability, and it can't produce another action potential. Was that really long or really short in skeletal muscle?
Really short.
Really short, because remember what we were able to do is we were able to stimulate that skeletal muscle cell really quickly and add a bunch of twitches together. What did we call that process? What did we call that process? Wave summation. Excellent. Wave summation. Perfect. Wave summation. And when we got wave summation to its peak, we were able to tetanize the muscle. Basically cause the muscle to produce this long sustained contraction. Now, is that something we would necessarily want to do with cardiac muscle? Do we want to tetanize our cardiac muscle where it's locked up in a contraction and can't stop?
No.
No. So notice, as we mentioned, where we have a very, very short refractory period, 1 to 2, or even just 1 to 5 milliseconds in skeletal muscle, the refractory period in cardiac muscle is over 250 milliseconds. Remember, the contraction only lasted 200. So if you think about it, the contraction is definitely going to be done before we can stimulate it a second time. So there's no way we can wave summate the heart. There's no way we can tetanize the heart because there's not going to be any tension left by the time it can be excited again. And here's the other big difference. Yes, when we talked about skeletal muscle, we talked about 3 types, right? Fast glycolytic, fast oxidative, and slow oxidative, but they all contracted the same way. Well, here in cardiac muscle, we have 2 very, very different types of cardiac muscle cell. The vast majority, 99% of the population, most of the cardiac muscle cells are what are known as contractile cells. And guess what contractile cells do? Not a trick question. There you go. Exactly. They contract. Excellent. Contractile cells contract. That's pretty darn convenient. But as we know, there's always those pesky 1%ers. And in this case, those pesky 1%ers are those special cardiac muscle cells that are actually autorhythmic. They are able to excite and produce their own action potentials. And those that can produce their own action potentials, as we mentioned, was what we call autorhythmic. So those are our autorhythmic cells, but what are also known as our conductive cells. So conductive or conduction cell is one type of cell. Contractile is a separate type of cardiac muscle cell. And so let's talk about those. First, we're going to talk about the contractile cells, because these are the ones that are most similar to our skeletal. And since we understand we've mastered skeletal muscle cells, that is going to be super convenient for us. So let's draw a nice pretty line right down the center. Hold on, I need to clear up some of these things, move them out of my way so that we can do this. So, fine, and we'll make it black, and we'll do it, do it here. Not quite halfway, but close. Excellent. And then we'll do the same thing like that as well. Perfect. I think I can make that work. All right. Let's start first talking about some things we already know and understand. Over here, we will talk about skeletal muscle action potentials. Excellent. Now, we know that we're going to have to draw a graph. So when we draw that graph, we always want to label our graphs. And in this case, our label is going to be time versus voltage. Excellent. Now, if I take a skeletal muscle cell out of your body, stick it in a petri dish, provide it with all the oxygen and the nutrients it's going to need, it is going to stay alive, and it is going to be very happy. Is it necessarily going to contract? Oh, I'm sorry, hold on real quick. Nabil? Yes, when someone needs a pacemaker, it's typically because there's problems with the autorhythmic cells. And we'll talk about the autorhythmic cells, and then we'll talk about how they go wrong. Excellent. It's not going to contract. It's going to be happy. And we're going to tell that it's happy because is its membrane potential going to change? Is the voltage inside that cell going to change? No, it's going to stay at some magical point. What was that magical point where it's going to stay at? What would we call that magical point? Where if we let it be, it would just happily, oops, hold on, wrong button. I need that one. I need that one.
Resting membrane.
Stay there forever. Excellent. We called that the resting membrane potential. And I don't have a lot of room here, but maybe if I do it small, sneak it in. Resting membrane potential. And someone remind me again what the resting membrane potential of a typical cell is?
Negative 70 millivolts.
Negative 70 millivolts. Excellent. Perfect. So that is our resting membrane potential. You are correct. Excellent. That is a good starting point. However, leaving things where they are is, happy for the cell, but it isn't very useful for us. We want it to do work. And as someone pointed out, to get it to do work, we have to get it up to that magical point. And as someone mentioned, that magical point that we need to get it to, and let's cheat with this and do it that way. There is some magical, mystical point we have to stimulate the cell up to. And what was that magical, mystical point we have to stimulate the cell up to?
Threshold.
Threshold. And when we get that cell to reach threshold, it is going to fire its action potential. So again, we'll use orange. We depolarize it to threshold. Let's make that a little bigger. Depolarize it to threshold, and then we get that rapid depolarization, followed immediately by a rapid repolarization back to its resting membrane potential. All right, we generate that massive action potential, the action potential, but it's very fast. As we talked about, it is going to last about 1 to 2 milliseconds. All right. Now, we know something else that we learned way back in 430. Oops, now hold on. I want that to stay orange. I want this new one to be black, to be 18. We know that when a skeletal muscle cell, again, we're still talking about the skeletal muscle cell, generates one, right, muscle action potential, that one muscle action potential produces one what? Well, one neural action potential produces one muscle action potential, and one muscle action potential produces one twitch. There you go. Excellent. So now we need to remind ourselves, oops, if I can spell, one twitch. Excellent. So what we need to talk about now is a skeletal muscle twitch. And again, remember that twitch is just basically the beginning of our contraction. We're going to add those together for the contraction. It's where we're generating tension. So we need another graph. And we don't want that to be green. I mean, the orange. All right, let's see. Pretty graph. Pretty graph. This is, of course, time down here. And then this over here is now tension. Oops, which apparently I can't spell. There we go. Excellent. Tension. So right here, at this point right, no, not what we wanted. On orange now. At this point right here is when we generated our muscle action potential. Remind me again, a twitch has 3 phases to it. What are the 3 phases of a twitch?
Latent period, contraction, and then relaxation.
Excellent. So those are, and I like that, so let's write them out. 3 phases. And those 3 phases were the latent, they were the contraction, and they were the relaxation. Excellent. 3 phases. And remember, all of these phases relate to changes in tension in the muscle cell. So that'll make it easy. Well, it is easy if I can spell phases correctly. Excellent. So, someone remind me how the tension changes during the latent phase.
No change.
No change. Excellent. And I'm going to elongate this a little bit to exaggerate it, but we have that latent period where there is no change in tension. How does the tension change during the contraction phase?
It rises.
Go, all right.
It rises, it increases. And how does the tension change during the relaxation phase?
Go down.
It increases.
It goes down until it's back to 0 again. So we have these 3 phases of our twitch based on where and how the tension changes. And I'm just going to abbreviate here: latent phase, contractile phase, right? Relaxation phase. And then, just to make sure we understand it, also right here at the end of it, we are now back at rest. Excellent. And as we already finished talking about, this entire twitch in a skeletal muscle cell is somewhere around 20 to 100 milliseconds, is about how long that process takes. All right. Questions on that? There really is no new information here on the board. This is all stuff you should have learned in 430 and hopefully remember from 430. We talked about skeletal muscle a lot. We talked about its action potentials. We talked about its twitch. So really, there's no new information here. This is really here just as a reminder. So hopefully you remember this. And if not, well, then I guess it's a good thing we spent a little time going over it. But what we need to do now is compare what we know, and that's what's going on with our skeletal muscle, to what's going on in our cardiac contractile cells. So here in our cardiac contractile cells, and let's make that, excuse me, a little bigger. No, not that big. Our cardiac contractile cells, and now we can talk about their muscle action potential. And I gotta move on my tiny windows over to the other side so I have some playroom. Excellent. Now that works. All right. We are going to start off the same way. We need a graph. So we need our X and Y axes. And again, here, down here, we will be doing time. And then here, up here, we will do voltage. And here's the cool thing. The process starts the same way. Our cardiac contractile cells have a resting membrane potential. So if you leave them alone, they will happily sit at their resting membrane potential forever. So if you leave them alone, they will happily be there. They'll stay there happily at the resting membrane potential forever. However, if they are stimulated, and in this case, remember, the stimulation comes from the gap junctions. If the cell next to this one spreads a whole bunch of positive ions, a big, huge positive signal to this cardiac contractile cell, then it is going to generate its muscle action potential. And the process is going to start the same way. So what happens is we get a rapid depolarization. Can you guys read that okay? I just want to make sure I have enough space in there. Is that too small, or is that, can you guys read that?
It's good.
Okay.
Is it rapid because they all shoot the signal to each other, so it basically all happens at once?
No, again, great question. No, just like before, what's going to happen is that the spread of the positive signal is going to get this cell to that magical point, and that magical point is going to be threshold. So again, this one's going to have a threshold. And when we reach that threshold, what's going to happen is basically the same thing that happened over here with our skeletal muscle cell. What causes our big rapid depolarization of a muscle cell? We can go back and write this over here as well. What causes our rapid depolarization in our skeletal muscle cell? Anyone remember? We open something. What kind of channels do we open?
Sodium channels.
Sodium.
And more specifically, voltage-gated sodium channels. We open a massive number of voltage-gated sodium channels. So we open a ton, I think is the technical term, of voltage-gated sodium channels. And we have a massive influx of sodium. Sodium is trying to reach its equilibrium potential. And anyone remind me what the equilibrium potential of sodium was again?
Plus 30?
No, plus 30 is, remember, what the peak gets to of an action potential. But the equilibrium potential of sodium was actually positive 66 millivolts. Oh. Excellent. So here, the same thing happens. Here, we get this rapid depolarization because, again, we are going to open a massive number of, and I'll just abbreviate it here since I wrote it on the other side, voltage-gated sodium channels. And we get a massive influx of sodium. And that sodium rushing into the cell makes the cell really, really positive. Exactly same process. So far, the two processes, what happens in our skeletal muscle, what happens in our cardiac contractile cell is the same. Anyone remember over here what makes the cell repolarize so quickly?
Potassium leaving the cell.
There you go. Voltage-gated potassium channels open. We get a massive efflux, outward movement of potassium, and the cell gets more negative. And that's why it goes right back down right away. But that's not what happens in our cardiac muscle. In our cardiac muscle, something really interesting happens. So let's actually change colors. Now, as we know, our voltage-gated sodium channels are going to inactivate. So no new sodium can come in. That's the same thing that happens in both of them. However, what happens here is we actually open calcium channels. And if we open calcium channels, which way is calcium going to come?
In the cell.
Calcium go into the cell or out of the cell?
In.
All right, calcium is going to enter the cell. Now, as we know, calcium entering the cell, calcium makes cells do wonky things. But remember, calcium is also a double positive ion. So calcium entering the cell from outside the cell makes the cell more positive. The other thing, interesting thing that happens during this time is that potassium channels actually close. All right? We have a fancy way of saying this. We say the permeability of potassium decreases. Permeability just means how much can cross the membrane. If we close potassium channels, then less potassium can leave the cell. And that's the key. When this happens, less potassium leaves the cell. Potassium is what type of an ion? Positive or negative?
Positive.
Positive. So if you have less positive ions leaving the cell, does that make the cell more positive or make the cell more negative?
Negative.
Well,
Positive.
again, if you have, think of it this way. If you have $100 in your pocket and you're normally on a Monday, you have to give out $10. But on this Monday, you only have to give $8. Right? You've given off less money. You have more money than you started with. You're a little bit more in the positive. And the same thing's here. If less potassium leaves, the cell stays more positive. So notice, sodium's not coming in anymore, but calcium is coming in, and potassium isn't leaving. And so those two things together, combined, they keep the cell depolarized for a longer period of time. And what ends up happening is we get this prolonged depolarization. I don't want that big. Let's go back here. We get this prolonged depolarization of the cell. And this prolonged depolarization we call the plateau phase. So we get this plateau. That's not right either. How do you spell plateau? None of those look right to me.
E-A-U.
E-A-U.
U?
A-T-E-A-U.
No, U's at the end.
U's at the end?
Yeah.
E-A-U? Okay, yeah, that looks right. Thank you. Excellent. Excellent. We reach this plateau phase where we get a prolonged depolarization. Excellent. Perfect. All right.
Phase or phase?
Phase.
Phase,
Apparently I can't spell phase either. I get one letter just like you guys.
yeah.
All right. Excellent. So we get this prolonged depolarization phase as a result of that. But eventually, and we'll use blue for this. Eventually, after a delay, voltage gated potassium channels open. And when those potassium channels open, potassium leaves the cell. And of course, potassium leaving the cell repolarizes the cell. And so the cell comes back to the resting membrane potential. And remember, as I said, how this one, the muscle action potential in a skeletal muscle cell lasts for 1 to 2 milliseconds. Remember here, we said that this muscle action potential can last over 200 milliseconds. So we have a much, much longer action potential as a result of this. All right.
Mm-hmm.
Questions on the muscle action potential? No. No, hyperpolarization occurs in the nerve cells, but it doesn't really occur in the muscle cells. So really, they pretty much just go back to rest. Great question, though. All right, excellent. So now, we have discussed a cardiac contractile cells action potential. Let's talk about a cardiac contractile cells twitch. Now, again, it's pretty much the same idea. One muscle action potential produces one twitch. So that's going to be the same. And it has the same 3 phases. So if we draw our graph here, and again, we are going to go with tension over here, and you're in my way. Time down here. And again, if right here is when our muscle action potential occurs, we are going to get the exact same 3 phases. We are going to get a latent phase, a contractile phase, and a relaxation phase. Right? Same 3 phases that we saw before: a latent phase, a contractile phase, a relaxation phase. All of those things are going to occur the exact same way. The only difference is the timing. Whereas here it was 20 to 100 milliseconds. Remember, as we talked about here, this single twitch is going to last around 200, is going to last around 200 milliseconds. Sorry, I got carried away here. Let's go back. This one doesn't last 200 milliseconds, but this action potential can be over 15 times longer than the skeletal muscle action potential. Whereas this is the one that lasts over 250 milliseconds. The twitch lasts over 200 milliseconds. And remember one more thing, and I'll do this one in black. Remember also, as we talked about here, I'll make it a little bit bigger so we remember. Here, our refractory period is going to be 1 to 5 milliseconds. Over here, our refractory period is going to be over 250 milliseconds. Longer than the twitch itself. So again, we can't sum it together. We can't tetanize our cardiac contractile muscle. All right. All right. I've done an amazing job of drawing this on the board. We don't have to worry about what the voltage of the plateau phase is. We're not. Notice I purposely didn't give you the resting membrane potential either, because they're slightly different in cardiac muscle, but I don't care about you knowing those numbers.
Will we post those pictures on the canvas?
Yes, I will save these and post them on Canvas, but as I will show you, as truly amazing as my art always is, we actually have the pretty pictures from your textbook that do a nice job of showing this as well. Notice here we see that skeletal muscle action potential, and in colors they've actually given us the absolute and relative refractory period of that, and it's very short. And there is the twitch that it produces. Notice the refractory period is less than the length of the twitch, which is why we can, we are able to tetanize it, we are able to sum it together. Here, we see the action potential of our cardiac muscle with that prolonged depolarization, that prolonged plateau phase. And again, as I mentioned, it's going to produce a contraction that lasts for a much longer period of time, basically past the refractory period. All right, and I think I have, again, it goes through the steps here, but I think the next slide does a nice job of showing this as well. Notice this cardiac muscle action potential has 3 phases to it. So I think I have that here. Yeah, here we have this here. Notice here we see those 3 phases. Phase 1 is that rapid depolarization. And again, remember, as we talked about, this is because we are allowing sodium into the cell. Here in the plateau phase, it's primary calcium entering, but remember also, we also stop potassium from leaving during this phase, and that gives us that prolonged depolarization, that plateau phase. And then finally, potassium is finally allowed to leave, and as the potassium leaves, it is able to repolarize back to rest. Absolutely, Jessica. This would make a great essay question. Not just, I could either ask you to describe the action potential of a cardiac contractile cell, or I could ask you to compare the action potentials of a cardiac muscle contractile cell and a skeletal muscle contractile cell. So absolutely, again, if you notice here, 2 definite essay questions. Usually when we have 2 processes, we have 3 questions. Describe process 1, describe process 2, and compare them, right? Understanding how the muscle action potential works isn't going to be an essay question that I would ask by itself, because you had to learn that in 430. So comparing them, or just talking about the contractile cells action potential, is absolutely a great potential essay question. Absolutely.
I have a question.
Yes.
So the resting membrane potential is the same as the skeletal muscle cells, right?
No. Notice here the pretty picture shows it's at 90. It is different. So if again, and as I mentioned, if you, not so unsubtly, I did not give you the resting membrane potential of the contractile cell. I did not tell you the threshold of it. I did not tell you what the peak was of this. Those are numbers that I don't care about you memorizing. Are there going to be numbers that you're going to need to memorize in this class and even in this section? Absolutely. But these aren't them.
Okay.
Okay.
And you said that contractile cells make up 99% of the heart cells, and then the other one is conduction.
Yep, exactly. So you're absolutely correct. If we've talked about the contractile cells, then what do we need to do next?
Conduction.
Conductive cells, absolutely. All right. Excellent. Can't figure out what I'm doing. Oh, there. Perfect. All right. And again, as we mentioned, the reason we have a much longer refractory period, excuse me, is because we don't want to wave summate. We don't want to tetanize our cardiac muscle. We want a long sustained contraction to make that efficient pumping, but we don't want to tetanize the muscle. All right. Questions on that? All right, excellent. We have done that. Now for the fun stuff. All right, I'll say this one more time just to make sure that I've done that. Perfect. So now I can clear it. We have talked about our cardiac contractile cell. But as we mentioned, we have that 1% of heart cells that are different. And these 1% of heart cells that are different are what we call the conductive heart cells, cardiac muscle cells. And again, what is special about them is that they are autorhythmic. Remind me again what it means to be autorhythmic.
They don't need signal. They
Based.
Yeah, they don't need a stimulus. They're able to produce their own action potentials. They can actually produce their own action potentials. So unlike skeletal muscle, unlike cardiac contractile cells, where, as I said, if we rip one of those cells out and put it in a petri dish and provide it with all the oxygen and the nutrients it needs, it will happily sit there at its resting membrane potential forever. It only contracts when it is stimulated. But this cell, if you pull one of these cells out and put it in a petri dish, it's going to happily produce its own action potentials on its own. It does not need any input from the nervous system, any input from hormones or anything else to cause it. It is able to produce its own action potentials. And the key reason for this is that, and I'll put it in capitals so that you know it's important, these cells do not have a resting membrane potential. Right? Instead, they have a cycling membrane potential. And remind me again what it means to cycle.
repeats again and again.
Yeah, it's a continuous process that goes around and around and around. Does that mean there's truly a starting point and an ending point? No, there isn't truly a starting point and an ending point. But if we're going to describe a cycle, we have to at least pick one point in time to start it from, but we know it's going to be continuous. All right, so let's do some drawing. The first part of the drawing is I'm actually going to draw, I don't want purple, our cell. Here's the cell. I'm going to make it a little elongated because I want some space to write and some space to play. And let's move it over a teeny bit more. There we go, excellent. And then I need a nice dividing, maybe put that there so I have a little bit of room to write around it. Actually, you know what? I think I need to make that cell smaller. I don't need it to be quite so big. That gives me more room to play. There, let's do that. Excellent. And then over here, we are going to be measuring our voltage over time. So again, time down here, voltage over here. Now, for this one's a little bit more complicated. So here's, for those of you who have not had me for 430, here is what we are going to do. Here on the board, all pretty, I'm going to go through this entire process. As we do this, what's going to happen is we're going to go through this process step by step by step by step. And for this, I strongly encourage you to just put your pens and pencils down, cross your legs, cross your hands, just sit, watch, listen, and think. Then when we're done with that, we will go through all the pretty pictures and all the pretty words a second time from the lecture. And at that point, you can furiously write away whatever it is that you want. But for this first time that we go through it, I really want you to just watch and think and listen so that it can make some sense to you. And then we'll be able to then go through it again and you can write it so that you can retain it. Okay? All right. Now, I've got my time, I've got my voltage, but I have an issue here. And as I've already mentioned, these cells don't have a resting membrane potential. So normally I have some point that I could put over here. Over here, that would be, that'll be okay. Yeah, we'll make that work. All right, I think that can be work. That's fine. We'll put it there. A resting membrane potential. But this cell doesn't have a resting membrane potential. But what it does have is voltage-gated channels. And if a cell has voltage-gated channels, while it may not have a resting membrane potential, what does it have?
Threshold.
Threshold. Excellent. It is going to have some threshold. All right, it is going to have some threshold to it, and that threshold is going to be its point. And let's go ahead and draw that threshold all the way across. And of course, as we know, the threshold is where our voltage-gated channels open. Threshold is that magical point where, and let's make this one smaller, where our voltage-gated channels open. Perfect. All right. Questions on that?
Sodium voltage-gated
Well,
channels.
I notice I didn't say sodium, did I?
No, you didn't.
No, I didn't. Excellent. Well, I didn't say sodium, and I didn't say sodium on purpose. Although, as we know, all big depolarizations are sodium, right?
Mm-hmm.
Of course, so that must be the case here as well. All right, let's find out. All right, excellent. So, again, we could start this process anywhere in this, but for argument's sake, I'm going to start us down here. So let's start with a dot, and let's make that dot purple. And I'm going to start down here below threshold. Okay? Now, here is the cell below threshold, and there's something interesting happening to this cell. This cell has, and let's put 3 of them here. These 3 channels here happen to be potassium channels. And what's interesting about these potassium channels at first is the potassium channels are slowly closing. So again, at the beginning of our process, it's not truly beginning of where we're describing it. What's happening right now is our potassium channels are slowly closing. We won't worry about what type of potassium channels they are. We won't worry about why they're closing. But for right now, let's just think of the fact of it. This cell is a normal cell in that there's a lot of potassium inside, very little potassium outside, a lot of sodium outside, very little sodium inside. So if I have 3 potassium channels opening, remind me again which way potassium goes?
Out.
Potassium leaves the cell. Excellent. So if I have this cell with 3 channels open and potassium is pouring out of it, and then suddenly one of these channels closes, what happens to the amount of potassium that leaves the cell?
Not as much leaves.
Not as much leaves. So less potassium leaves the cell. And if less positive potassium leaves the cell, What effect does that have on the cell?
More
Cell
positive.
stays positive.
The cell's going to stay more positive. Again, is it going to be a rapid process? No, but it slowly gets more positive. Remember, these three potassium channels that I've put on here are not the only channels on the cell. There are leak channels and pump channels and all those things. Remember, when we talk about a cell being at its resting membrane potential, it's not that no ions are moving in and out, it's that there's no net change. Well, here we're changing. Everything is normally moving in and out, but less potassium is leaving. And if less potassium is leaving, then overall, the cell slowly gets a little bit more positive. And in fact, that's what we call this phase. This phase is what we call the slow depolarization. All right? And we can draw that. The cell is slowly getting more positive, slowly, 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 until something special happens. What's that special thing that just happened?
Reaches threshold.
Reach threshold.
Excellent. The special thing that happened is that we reach threshold. So the next thing that happens is we reach threshold. The cell reaches threshold. And when the cell reaches threshold, of course, as we know, the voltage gated channels are going to open and ions are going to be able to flow. Exactly. And of course, this is going to give us a massive, rapid depolarization. And this, of course, is our action potential. Right? We're going to end up getting this big, massive, big, rapid depolarization from opening these voltage-gated channels. And of course, what type of voltage-gated channels are we going to open to get this big, huge, massive, rapid depolarization that is our action potential?
Real?
Exactly. Calcium channels.
Calcium.
Wait, what?
You said calcium or sodium?
I said calcium. I even wrote
Okay.
calcium. That can't be right, can it? Yeah, it's calcium. Every other action potential we have ever talked about in the history of 430 and 431 has involved sodium, except this one. In this case here, what actually happens is we actually open these voltage-gated calcium channels, and we get this big, huge, massive, rapid depolarization. So we had the slow depolarization, now we have the rapid depolarization.
So I want to ask about slow depolarization.
I'm sorry, say that again?
I would like to ask about the slow depolarization.
Absolutely.
So
What would you like to ask?
Slow depolarization in when not all potassium channels open. So some of them must be closed.
Well, no, remember, the reason we're getting a slow depolarization is because potassium channels are closing.
Okay.
So first one potassium channel closes, a little less potassium leaves, and the cell gets a little bit more positive. Then a second one closes, and the cell gets a little bit more positive. And then a third one closes, and it gets a little bit more positive, right? So we're closing all of these potassium channels, and let's go ahead and draw one more. Oops, no, hold on. I want that, I want that. There we go, put another one right there. Oh, that's gonna be over the top of it, is it? Yeah, that doesn't work. All right, never mind. You get the idea. So erase that and erase that. Perfect. So all that's happening during the slow depolarization is that we are getting less potassium leaving because potassium channels are closing. And I know it's a little tricky because I haven't told you what kind of potassium channels and I haven't told you why they're closing, but we're about to get to that. All right. They don't all close, but most of them are going to close. most of them are enclosed, but not necessarily all of them. Again, I've only drawn 3, so it's not easy. But remember, this cell is going to have hundreds of them. So will all 100 close? No. Just enough so that it gets positive enough to get to threshold. And again, at threshold, and now we can change our drawing over here to now put our orange ones at threshold. And again, notice I'm putting these orange channels on one side of the cell and the purple on the other. Is that how it's organized? All of one channels are on one side and all the other ones are on the other? No, I'm just doing this for a simple schematic to make this simple and easy to show how this is going to work. These are evenly spread throughout the entire cell. But here we have those voltage gated, calcium channels. And that's going to be too big, so let's make that there. And these are the ones that open at threshold. And of course, when they open at threshold, massive amounts of calcium enter the cell. Perfect. And we get our rapid depolarization. All right. Questions on that? All right, I know I half blew your mind with voltage-gated calcium channels columns in the action potential. Here's the other half of that. Calcium makes cells do wonky things, doesn't it? Well, in this case, we have a large amount of calcium inside the cell, and let's draw that calcium as some little orange balls. Now, as we know, does the cell like having calcium inside of it?
No.
No. So what's one of the ways that cells get rid of calcium inside of it? If a cell has calcium inside of it and wants to get rid of it, how could it do that? Pumps. Excellent. We have these pumps, and these pumps are going to use ATP, and they're going to use ATP to kick calcium out. So we're going to have some calcium pumps. And those calcium pumps are going to remove calcium from the cell. But if I have all these voltage-gated calcium channels open, is this pump going to be able to do anything about all this massive amount of calcium coming in?
No.
No. So we are going to get an increase in calcium inside the cell. So then the question becomes, what is it that this calcium does? Well, let's go back to these purple channels we talked about. At the beginning, we mentioned these purple channels, and we said that they're calcium channels, but it turns out these are chemically gated, or remember the other fancy word we can use is ligand gated channels. Remind me again what that means to be chemically gated or ligand gated.
Doesn't it take a hormone to open the channels?
Well, it isn't necessarily that it has to be a hormone, but you got the right idea. Some type of chemical, some type of molecule has to bind to it to open this channel up. And guess what that chemical might be? Sometimes it's a protein, but it doesn't always have to be a protein.
Sodium?
Some kind of chemical, some kind of molecule has to bind to this channel. And guess what that molecule is not even the right word. That chemical is calcium.
Is it acetylcholine?
Nope, it is calcium. This is a calcium-gated potassium channel. And again, let's make that a little bigger so we can see that. Calcium-gated. What that means is that calcium is the key that is going to unlock the door. Calcium is going to come over here and bind to it. And when calcium binds to these channels, what happens to the channels? Oops. Undo.
Shape and open.
They're going to open, absolutely. So let's grab that and move it out of the way, and grab that and move it out of the way. And notice when that calcium enters the cell, now what happens is the calcium binds to the channels and opens them. And when it binds to the channels and opens them, these channels open. What happens when these channels open?
Potassium goes out.
Potassium leaves the cell. And when potassium leaves the cell, what happens to the cell?
Repolarizes.
Polarize.
Yeah, it gets more negative, absolutely. And so that is exactly what is going to happen. What is going to happen is that calcium is going to bind to the potassium channels. And as the calcium binds to the potassium channels, so let's write this out. Calcium binds to the potassium channels. The potassium channels open. As the potassium channels open, potassium leaves the cell. And the cell gets more negative. Now, the word that we actually use here is we say the cell repolarizes. Now, when I say the cell repolarizes, what does that term usually mean, to repolarize? What does it mean to repolarize?
To go back to the resting membrane.
Go back to the resting membrane potential. But does this have a resting membrane potential?
No.
No. So saying that it repolarizes is a little bit of a cheat, but it is the term is commonly used that way, so I'm okay with you saying it. I'm okay with you saying that it is going to repolarize, although technically it just gets more negative, but repolarizes is a good thought. All right?
What about the calcium? It was a lot of calcium inside of the cell.
True, a lot of calcium came in, but then remember some of it's being pumped out, and now more potassium is leaving. Think of it this way. The more calcium that comes in, the more potassium channels you can open, and the more potassium that is going to be able to leave. Okay? And so what happens here is we get a rapid repolarization. All right? Questions on that? The actual timing of these, we're talking about fractions of milliseconds, milliseconds and fractions of milliseconds. So it's not, the timing isn't quite as important, but it is fast. It is a rapid repolarization, similar to the speed of the depolarization. All right.
So could you say that the refractory period is kind of just when the potassium is like leaking out of the cell?
But remember, these don't have a refractory period the same type of way. These don't have a resting membrane potential, so these cells are different. They don't have a refractory period in the same way that the other ones do, because they're constantly changing, constantly in flux.
So they just go back to slow depolarization, correct?
Right. Well, yeah, exactly. We're going to finish the process off. Remember, it's a cycle, so we have to continue. It's not going to stay here, right? This is not a resting membrane potential. This process is going to continue. It's a continuous process. And remember, a refractory period is how soon a cell can be stimulated again. This cell doesn't receive stimulation. It produces its own action potential. So it doesn't have a refractory period because it's not irritable, because it doesn't receive stimuli. It's making the stimuli. It's the one that stimulates everything else. Okay.
So it's just kind of always going.
I'm sorry?
It's just always going. Like,
yeah, it's continuous. Because watch, let's finish the process because we're not quite done. But Ryan's kind of hit it on the head here. Notice something interesting happened, and let's go ahead and use red for the color of this one. Right here, something interesting happened. Right here, we passed threshold on the way down. So right here, our cell passes threshold on the way down. And what happens when we pass threshold on the way down?
Channels close.
There you go. Our voltage gated calcium channels are going to close. Remember, hyperpolarized means to go below the resting membrane potential. So remember, we don't have a resting membrane potential here. This red line is threshold. That's not a resting membrane potential. So because we're going below it, it's not considered a hyperpolarization. It's threshold that we're going past. All right, but you guys have the right idea. Our voltage-gated calcium channels are going to close, and when they close, no new calcium can enter the cell. So once these channels close, oops, no. Once those channels close, no new calcium can come inside. However, there's still a lot of calcium left inside. So do our potassium channels close right away?
No, they slowly close.
Yeah, there's still a lot of calcium inside, but now our pumps are going to be able to move it out. So what happens is our pumps are now able to start removing the calcium inside the cell. And as there's less potassium inside the cell, less calcium, as there's less calcium inside the cell, our calcium gated potassium channels start to close. And as our calcium gated potassium channels start to close, what starts to happen to the cell?
Slow depolarization.
Right. Less potassium leaving the cell causes a slow depolarization, and the process continues. So now we are getting that slow depolarization. And when it slowly polarizes to threshold, what's going to happen then?
Rapid depolarization.
Rapid depolarization.
And then what happens?
Rapid repolarization.
Rapid repolarization, and the process continues. Excellent. And there you go. Just that simply, we have described the activities, the action potential of our conductive cardiac muscle cell. Right? It doesn't have a resting membrane potential. Its membrane potential is constantly changing because of this constant cycling of potassium and of calcium. And notice it's potassium and calcium that is driving this process. All right. Questions on that?
Does it change with heart rate?
Great question. So yes, so let's talk about that. Let's talk about that in a second. Let me answer Levon's question first. Levon, remember that our cell always has ATP in here. There is always going to be ATP here inside of the cell. And that ATP inside of the cell is going to help us to pump calcium out. The example I like to use for this, though, is like if you're in a rowboat. If you're in a little rowboat, and while you're in the rowboat, someone drills 75 holes into the bottom of your boat, and you have a little hand pump. You can use that hand pump to get water out. But are you going to be able to get it out as quickly as that water can come in? No. And so what happens is the boat fills more and more with water. And that's what's happening here. that pump is kicking calcium out all the time. But so much is coming in that it's not having an effect. However, if you can plug those 75 holes, so no new water is coming into your boat, now your little hand pump can make a difference. Now it can get rid of the water so that you get rid of it in there. And that is how this is working. Thank you. I like that too. So that's it. So it's doing it all the time. But here, when they finally close, is when it can make a difference. All right. Excellent. So now let's answer the next question. Alex, were you the one who asked this?
Yes.
Okay, perfect. So this is our normal heart rate of our cell. In fact, as I mentioned, I ripped that cell out of your heart, that conductive cell out of your heart, put it in a petri dish, give it all the oxygen, the nutrients, and it's going to keep on this cycle. And every action potential basically causes a heartbeat, right? So if I sat here and watched it, what had happened is it would produce 100 of these action potentials in 1 minute of time. That would lead to 100 beats per minute heart rate. Is anybody's heart rate 100 beats per minute right now? I know it's been a while since we've taken a break, so probably, hopefully nobody's had, their quad shot from Dutch Brothers or anything like that. No, nobody's heart rate should be beating that fast. The reason for that is because this process can be modified. Remember, one of the keys we talked about back in 430 is that the heart is autorhythmic, but it can be modified by our autonomic, let me do it this way, nervous system, and by hormones. And the way it does that is by opening new channels. Both our autonomic nervous system by releasing neurotransmitters. So for our autonomic nervous system, let's put in parentheses here, neurotransmitter. Oops. Neurotransmitters. And by hormones, we can open new chemical signals. Excellent. So let's take the example of while you're sitting here at rest. While you're sitting here at rest, your heart rate is lower than 100 beats per minute. Where am I going to put this? I think I can sneak it in here. Let's see if I can sneak it in up here at the top. So let's do this first. All right. Which branch of our, it should be somewhere between 60 and 80. It could be up to 100. Again, it depends on the person, but usually 60 to 80 is a good range. So which branch of our autonomic nervous system is responsible for lowering heart rate?
Parasympathetic.
Excellent. It is the parasympathetic. And because you had an amazingly excellent instructor for 430, what neurotransmitter does our parasympathetic nervous system release to our effectors?
Acetylcholine.
Excellent. Acetylcholine is the neurotransmitter that is released by the parasympathetic nervous system. What that acetylcholine does is it actually opens chloride channels. So let's go back to our purple dot here as our starting point. But let's add something new. And that something new we're going to add is a third channel. That's not green. Is a third channel. And this third channel, our acetylcholine can bind to. And when it opens it up, it's a chloride channel. So it's an acetylcholine gated chloride channel. Chloride channel. So when the acetylcholine opens it, which way does chloride go? Into the cell. Excellent. Chlorine Chloride ions are going to enter into the cell. So notice our starting point again was right here. Right? This is our starting point right here. There you go, exactly. I'm closing potassium channels unless potassium is leaving, but now chloride ions are also coming in. So if chloride ions are also coming in during this time, is it going to reach threshold faster or is it going to reach threshold slower?
Slower.
Well, as Ryan pointed out, it is going to take it longer to reach threshold. Of course, once it reaches threshold, we'll get our rapid depolarization, and then we'll get our rapid repolarization, and then it will start anew. But notice, because we've opened those chloride channels, it has taken us longer to reach threshold, and the heart rate slows down. Excellent. That one makes sense?
Yes.
Perfect. Let's do the opposite. If we want to increase heart rate, what branch of the nervous system would be responsible for that?
Sympathetic.
Sympathetic. Excellent. And what neurotransmitter does our sympathetic nervous system release to the heart?
Norepinephrine.
Norepinephrine. Excellent. Excellent. Norepinephrine. And that norepinephrine opens sodium channels. So now we have a new channel. This new channel is a norepinephrine gated sodium channel. And when that norepinephrine binds to it, which way is sodium going to flow through this channel?
Into the cell.
Excellent. So sodium is going to rush into the cell. Sodium enters the cell. Now, potassium's not leaving the cell. Let's go back again to our starting point. Here, calcium is being pumped out. Our our calcium-gated potassium channels are closing. Less potassium is leaving the cell, but now I've opened sodium channels, and sodium channels are rushing into the cell. And if sodium ions are rushing into the cell, is this cell going to depolarize slower or faster?
Faster.
Faster. It is going to depolarize faster, reach threshold faster, and then once we reach threshold, we get that rapid depolarization, we get that rapid repolarization, and then again, we increase the speed at which it reaches threshold, and then we get that rapid depolarization, and we get that rapid repolarization, and notice the heart is beating much, much faster than it was before. So you're right, Ryan, there are 4 different types of channels here, but notice 2 of them, the voltage-gated calcium channels and the calcium-gated potassium channels, they're the ones that set the normal pace. And it's the chloride channels and the sodium channels that modify it. And so in this way, when you're sitting calmly in the chair, you're getting more parasympathetic input into your heart. So you're opening more of those chloride channels and it's slowing down. When you have a 10-minute break and you've got to run down to the corner to get Starbucks and run all the way back, then you're getting more sympathetic input, more sodium is entering in the cell because that norepinephrine is opening up those channels and the heart beats faster. So notice not only here were we able to see how you establish that normal autorhythmicity of the cell, how having no resting membrane potential allows it to continuously produce action potentials, but we've even seen now how we can modify it, how we can make it go faster, how we can make it go slower. All of those things, carbon dioxide levels, oxygen levels, those are probably the two main things, although glucose levels could be affected. Metabolic wastes, an increase in metabolic wastes, an increase in temperature, all of those things are things that can modify the heart rate. Right, and again, we just talked about the nervous system, but remember, we also talked about hormones, right? What about adrenaline? Can adrenaline bind to those same norepinephrine receptors, let that sodium in and make your heart beat faster? Yeah, absolutely. So other hormones can affect and modify these things as well. All right. AFib is something different. Right now, we're just looking at one cell and seeing what's happening with one cell, but absolutely, Levon, you've got the right idea. These cells don't truly work by themselves. They work in a circuit, and that's actually what we're going to talk about next. We're going to talk about that circuit next. Great question. So the question is, can you trick your heart rate to go slower? Like trying to control it. I wouldn't say that it's necessarily tricking, but what we can do is try to take advantage of what we understand about sympathetic and parasympathetic innervation, right? Our sympathetic innervation is that fight or flight stress response. So if you closed your eyes and really spent 5 minutes really thinking about what it would be like giving a presentation to the entire school not wearing any pants, and you really thought about what that would like and sat there and tried to visualize it, do you think that might cause your heart rate to go a little bit faster as a result of that? Yeah, absolutely. In the same way that if we sat here and we tried to clear our minds of thought and we just sat here and said, and we were sitting in a lotus position and everything was quiet around us, could we slow things down to a point where could we enhance our parasympathetic input to our heart, like in meditation, and cause our heart rate to slow down? Yes. I'm not telling my heart to beat slower. You can't do that directly. But what you can do is by getting yourself in a more calm state, you can change that autonomic tone so that there's more parasympathetic than sympathetic, by clearing your mind, by stopping to worry about things. And that, as a result of suppressing the sympathetic and enhancing the parasympathetic, you could get the heart to beat faster. Yeah, there's some mechanical ways like the Valsalva maneuver where you can kind of cause it to try to beat slower mechanically and stuff like that. But again, that's usually not the best. We'll also talk about the Valsalva maneuver in as it relates to defecation as well when we talk about that. All right. All right, that went long, but that's okay. So here's what we'll do. This is a perfect stopping point. Let's go ahead and take our second break here. And then when we come back, as promised, we'll go through this whole process again with all the pretty pictures and all the pretty words on, from your textbook and from the lab slides to make sure that we understand this. And then, as Levon pointed out, we'll then put this single cell into the circuit of all the cells that, again, regulate the activity of our heart. All right, so this is a good stopping point. We'll come back and then we can review this again. This will give this a chance to sink in. Looks like it's 2:40. So where can I write this that won't take up too much space? I'll sneak it down here in the corner. Let's return at 2:40, so that's 2:55. Oops. 2:55, and at that point, I will start the recording. All right. Any questions before we take our next break? Excellent. Stunned silence. I love it. All right. See you guys back here in 15 minutes. Zoom the recording. And again, we can get rid of this scary picture now and go back to the slightly less scary, although I thought I did an excellent job of that. I'm going to this here and talking about our conductive cell, or that autorhythmic cell. Again, they only make up 1%. And as I mentioned, the big key to them is that they don't have a resting membrane potential, right? If you think about it, there are really two things we talked about this cell that are really special about it. They don't have a resting membrane potential, and their action potential isn't produced by sodium. All right? So, as we mentioned, there are really 3 phases to this. Why did those both come up? All right, I don't know why both of those came up at the same time. Let's pretend that we don't see that other one. Here, I'll cheat and put a big box over the top of it. The first thing that we have is that slow depolarization of potassium. Again, our potassium permeability is decreasing. And now we know why. The why of that is that our calcium-gated potassium channels are slowly closing, right? Those pumps can't remove all the calcium at once. So as they remove, 20% of it, 20% of the potassium channels close. As they remove 50%, 50% of the potassium channels close. So that's why we get that slow depolarization. Now remember, we can let sodium in, and if we let sodium in, this is gonna go, it's gonna reach threshold faster. Remember also, we can let a chloride in. And if we let chloride in, it's going to go slower. So remember, using our neurotransmitters, using additional channels, we are able to modify this rate. This is the part, the slow depolarization. How fast does it reach threshold? This is the part that we can modify. This is what we modify to change heart rate. How quickly the calcium channels open, how quickly the potassium channels open, that we really can't change. So the rapid depolarization and the rapid repolarization pretty much stay the same. What makes the heart beat faster and slower is how quickly or how slowly we reach threshold. All right, that makes sense? I guess the two ding dings mean yes, excellent. So second stage, we get that rapid depolarization from reaching threshold and having those voltage-gated calcium channels open. The voltage-gated calcium channels open, and we have a massive influx of calcium. But all that calcium coming in then leads to the opening of those potassium channels. Our calcium-gated potassium channels open. Wait, why is that? That's wrong. Hold on. Not sure why it says close there. Does it say close on your notes too? Make sure you fix that.
It says a rapid depolarization under number 1. On reaching decrease of K plus. I read it a couple days ago.
So we get that rapid depolarization from the calcium-gated potassium channels opening. That causes that rapid repolarization. But once we pass threshold on the way back down, those calcium channels close. And when those calcium channels close, the potassium slowly stops leaving. We get that slow depolarization, and the process continues. All righty. And again, this is a cyclical process. Notice we could have started this description at any point. We could have started at threshold, if that makes more sense to you. You start at threshold, you open the voltage-gated calcium channels, calcium rushes in, it opens voltage-gated, calcium-gated potassium channels, potassium leaves. As we cross threshold, the calcium channels close, and then we get the slow depolarization and it starts again. I don't care where you start the cycle, as long as you give me one complete cycle, you remind me that it continues. So however that works best for you is totally fine with me. All right. Questions on that? All right, excellent. So this is all happening in one cell, but as we mentioned, they're only 1%, but that 1% of the cells aren't just randomly distributed throughout the heart. They are interconnected and work together to form a system that we call the cardiac conduction system. And here we have the pretty picture from the textbook. Oh, in just a second that we will talk about and see. Now, one of the important things to remember is that every cell of the cardiac conduction system is autorhythmic. All right, every single one of them. However, that being said, they do not all fire at the same rate. Some are faster and some are slower. They form different structures in the heart, like the sinoatrial node, and the cells of the sinoatrial node are the ones that fire the fastest. Those are the ones that we talked about before that can fire a rate of about 90 to 100 times per minute, which is more than what you need while you're sitting here in the classroom. Right. Conversely, our second major structure is a structure called the atrioventricular node. It can produce heart rates at a rate of about 40 to 50 per minute. Could you survive with that as a heart rate?
Yes.
Yeah, absolutely. You could survive with that as a heart rate. Now, would you have a hard time carrying bags of grocery up 3 flights of stairs if that was your heart rate?
Yes.
Yes.
Yeah, absolutely. Absolutely, you would have trouble with that, but you could survive. However, there are other structures formed by the cardiac autorhythmic cells that can beat about 20 to 40 times per minute. And those aren't enough to maintain a viable, healthy heart rate. Yeah, that can't keep us alive. And so as someone mentioned earlier, when there are problems with these autorhythmic cells, and more particular with this cardiac conduction system, luckily we can modify it artificially with an artificial pacemaker. All right. There we go. Now, again, as we talked about, the sinoatrial node is the portion of the cardiac conduction system that is typically referred to as the pacemaker of the heart, and it's the one that is primarily responsible for setting your normal heart rate. And why would it be the one that is primarily responsible for setting your normal heart rate?
Beats the fastest.
Yeah, it's the one that beats the fastest, absolutely. So it's the one that tends to drive the other ones. Of course, as we already talked about, it would just happily beat away at 100 beats per minute. And we don't want our heart to beat 100 beats a minute all the time. There are times, like when we're exercising, we want to, and even faster. But there are also plenty of times when we want it slower as well. So we can use things like what? What are some of the things we can use to modify our heart rate? Come on, guys. I know the answer to these.
Hormones? Or
It
your
hasn't even been a
nerves?
long day yet. This is, yes, say again. Excellent. Hormones. Excellent. Hormones are one of the things we can use to modify it. What else? Like adrenaline. Absolutely. One of the big examples of that is adrenaline.
Stimulants.
Right. Absolutely. Some type of stimulants. Things like nicotine. Right, is a good example, or other type of recreational drugs could have that type of effect. What else? What's another stimulant that can increase your heart rate?
Caffeine.
Caffeine, there you go, excellent. All right, caffeine. What else?
What about sport?
Say again?
Sport?
Right, so if you're, yeah, absolutely. And so, but again, what is it that directly modifies it in that case? If you're exercising or if you're scared, it could be hormones, but what else could it be? You guys are overthinking this.
Sympathetic?
There you go, our autonomic nervous system, absolutely. And you are correct. We can have our sympathetic, that is going to typically increase, and our parasympathetic, which is going to primarily decrease. the rates of that. Absolutely excellent. All right. And so using things like toxins or medications or other types of stimulants, hormones, nervous input from our autonomic nervous system, these are all ways that we can modify our heart rate. Because as we mentioned, having a heart rate of 100 beats per minute isn't a good thing. Or is it? If I were to take your heart rate, or if you had a patient came in and their heart rate was 100 beats per minute, is that a good thing or a bad thing?
Depends.
Depends on if they're running.
There you go, exactly. That is the key. It's not like 100 beats per minute is a bad thing. It totally depends on the activity that you are participating in. If they have been meditating for 25 minutes and their heart is beating 100 beats per minute, then something's wrong, right? If, on the other hand, they've been on the treadmill for 20 minutes or they're in pain, those are cases that it could do that as well. And that's really the key to these two terms. I make a point of emphasizing this here because we think in terms of irregularities in the heart rate of being bradycardia and tachycardia. If someone is bradycardic, what does that mean?
Heart rate's too slow, below 60.
True, it could mean below 60, but again, it is dependent on the situation. Bradycardia is a heart rate that is lower than expected. Again, if somebody is sprinting on the treadmill and they've been sprinting on the treadmill for 5 minutes and you take their heart rate and their heart rate is 80 beats a minute, do you say, oh, that's above 60, that's totally normal? No, of course not, right? That would be incredibly lower than expected for that situation, right? Again, you would expect their heart rate, if they're sprinting, it could be 140, 150 beats per minute if they're running really, really hard. Now, if they're sitting calmly in your class and their heart is racing at 150 beats per minute, then they would be tachycardic, right? So again, bradycardia and tachycardia are lower than or higher than heart rates than expected for the situation. So it really is situational. You can't just give a flat number, right? 100 is tachycardic. No, it doesn't mean that. It means it's all related to the situation and the condition of the person at that time. All right?
Is it relative to their resting heartbeat?
Definitely, because again, people do have different resting heart rates. Someone, again, it's a great example. Someone can come in, you can have a patient come in who has a heart rate of 85, and that doesn't seem bad at all. But then you look back in their chart and you see the last 10 times they've been in, their resting heart rate was 62. If their normal resting heart rate is closer to 62 and they're sitting in your chair at 85, something different is going on, right? Absolutely. Right. So yeah, so a lot of it is related to what their normal heart rate is. As we talked about, we can have things like ectopic pacemakers. Those are things like we talked about, like caffeine and nicotine that can artificially increase the activity. And as we also talked about, if there's problems with the conductive system, we can actually have artificial pacemakers to help to regulate the activity of these conductive cells. All right. But let's talk about the normal system first. Here we have the pretty picture from your textbook that does a good job of showing us the different components of this. And here we see the components in a nice, pretty heart, the accurate looking heart. But as we know, I like to draw, so let's draw my heart. All right, I'll say this one more time to make sure we have it, and then I can go ahead and clear that. And again, because I'm so terribly, incredibly lazy, we will just draw the heart as a nice big circle. Because again, my goal isn't to draw this in an anatomical way. The goal here is to draw a schematic of this that is going to help us to appreciate and understand this more simply. All right. Now, as we mentioned, there are 5 main components. The first component, as we mentioned, is the sinoatrial node. All right, we need that to be black. We need it to be bigger now. The sinoatrial node. The sinoatrial node is located within the right atrium. And we'll cheat and go back to the picture for this in just a second. It's basically in the armpit of the superior vena cava. So let's cheat and go back to the picture from the textbook real fast. Notice here it is right here. I'll use the highlighter on this one. This is it right here. Notice right underneath the superior vena cava, right underneath there, kind of in the armpit of the superior vena cava in the right atrium is that sinoatrial node. As we talked about, the sinoatrial node is our pacemaker because it has the fastest firing rate. We start with all the cells at rest. And again, they're not at rest, but all the cells repolarized under threshold. And these are the ones that reach threshold first. These are the ones that are going to fire their action potential. And when they fire their action potential, that action potential is then going to spread across both atria. Again, thanks to those gap junctions, we are going to be able to spread this across the entire atria. But remember, we said in that cardiac muscle, the myocardium is not just cardiac muscle. There's also that fibrous skeleton. And the other thing we talked about that fibrous skeleton is, in particular, that fibrous skeleton is thicker where the valves are. And remember, the valves are all kind of found on this border between the atria and the ventricle, right? Think of it this way. If you stand, because again, remember, one of the things we talked about in 430 is electricity and water move in very much the same fashion, right? If you take a big rock and throw it into your pool, it's going to produce a wave, and that wave is going to spread across the entire surface of your pool. However, if you have one of those pools that have the hot tub connected to it but separate from it, if I throw a wave in the hot tub, it produces waves in there, but are those waves going to spread to the rest of the pool? No, and that's what happens here. Because of that fibrous skeleton stops the spread of the signal to the ventricles. So this is going to spread across all the atria, right? Our signal spreads across the atria. But doesn't travel down to the ventricles. Luckily, we have a second structure. And that second structure is the atrioventricular node. And guess where the atrioventricular node is located?
Ventricles.
Well, atrioventricular, so is it just in the ventricle?
Between the atria.
Yeah, it's going to be between the atria and the ventricles, absolutely. So it's basically located right about here. Well, let's not do that. Let's do this again. Here is our atrioventricular node. And so some of those action potentials that have been spreading across the atria are going to spread to the atrioventricular node. There's something really interesting about the atrioventricular node, right? If you notice, as we've talked about it, for the most part, these conductive cells are kind of a cross between the nervous cells and muscle cells. They're kind of like a combo plate. They don't produce contractions. They conduct electrical activity. So they're kind of like nervous cells that way. And what's interesting about these cells of the atrioventricular node is the cells are very small and very thin. Why might that be significant? What's the significance of these cells being very small and very thin? I don't know either, so let's just move on. Or not, come on. All right, excellent. Let's think of it this way. Back in ancient times, we used to be able to go to restaurants. And back in those ancient times, one of the fun restaurants to go to Mel's Diner, right? They have the great music, decent food, and one of the things, especially the thing that my daughter loves about it so much, is you can get a shake there. And when you get a shake there, not only do they pour it in the glass, but they give you the metal cylinder that they gave it to you in. So you get, it's like getting 2 milkshakes for the price of 1, and she absolutely loves it. And when they bring you that great, big, thick shake, do you want to grab like a coffee stirrer to suck that shake through? Or do you want one of those big, huge, like McDonald's is great that way. McDonald's has those big, huge, massive straws for a shake, right? Why do you want the big round shake? Because it has more space inside. Less of the shake is in contact with the surface of the straw, so you have less resistance and you have more flow, right? And here, you're absolutely correct. We get a slow conduction because we have the exact opposite. Since we are dealing with small, thin cells, what we get is a very large resistance. And that large resistance slows the spread of the action potential. Why would we want to slow the spread of the action potential?
Because you want to slow our heart rate, right?
Part of it is that, but let's think of it this way also. Remember, when this action potential is spreading across the atria, the action potential is spreading to the contractile cells. And when contractile cells are stimulated, what do contractile cells do?
They twitch.
They contract, absolutely. And when they contract, that pumps the blood. Now, if we are going to turn our heart to an efficient pump, do we necessarily want the atria and the ventricles contracting at the same time?
No.
No. So notice by slowing the signal down here in our atrioventricular node, and I should have put a 2 in front of that, so I'll cheat and do that there. This gives the atria a chance to contract before the ventricles contract. So we're able to slow the signal down, and it makes for a more efficient process. Our atria can contract before the ventricles can contract. And that's going to get us a much more consistent, much more efficient contraction. Now, connected to the atrioventricular node is a structure that is known as the atrioventricular bundle, or what is also referred to as the bundle of His. And of course, why is it called the bundle of His? Not your question. Why is it called the bundle of His?
Because it connects.
There you go, exactly. The reason it is called the bundle of His is because good old Bob His was the first one who discovered it, planted his flag in it, and got to be able to, put his name in it that way. Absolutely. All right. Excellent. In this case, that bundle of His is a large structure that carries the action potential from the atrioventricular node into the interventricular septum. And I'm going to have to cheat a little bit and draw us a second line. Nope, not purple. We have this massive muscle between the ventricles. And of course, as we know, that massive muscle goes up the sides on the right, and it's even bigger on the left. So let's crazily exaggerate it. And so here we have this atrioventricular bundle, or the bundle of His, and basically it carries the action potential into the interventricular septum. the big chunk of meat between the two ventricles. This bundle of His happens to be the location where most of our heart blocks occur. So one of the things I don't know if we'll get a chance to talk about is that we'll get a chance to talk about heart blocks, type 1, type 2, and type 3. Those heart blocks that occur typically occur here in the bundle of His, because this is one of the most narrow regions for it. Now, once it enters the septum, then that bundle of His branches. And as it branches, it forms multiple branches, which, not surprisingly, are referred to as the bundle branches. Those bundle branches carry the action potential to the apex of the heart. And then from there, conductive cells carry that signal up the walls of the ventricle. And these cells that carry the signal up the walls of the ventricle are what are known as Purkinje fibers. Some of you may have remembered the Purkinje cells we studied in 430 in the nervous system. Same anatomist. Again, none of these are axons because remember, these are conductive cells. Conductive cells are nervous-like muscle cells, but they are muscle cells. All right. And of course, if these Purkinje cells are carrying the action potential up the walls of the ventricle, then of course, that is what stimulates the ventricles so that they contract.
So the AV node doesn't tell the ventricles to contract, it just tells the atria to contract?
Well, the sinoatrial node is what tells the atria to contract. But it sends the signal to the atrioventricular node. It's the atrioventricular node who takes that signal and sends it to the ventricles. And then the bundle branches and the Purkinje fibers are the ones that then tell the ventricles to contract.
Okay.
Yeah.
So the AV node just kind of passes it on?
Yes. And again, slows it down. The real key to the AV node is that it slows it down. It's that toll booth that slows everything down so that we can get a complete contraction of the atria before we get a contraction of the ventricle. All right. Now, I do have one more question. This does seem almost sillily elaborate. Why not just spread the action potential down and across the entire heart? Why instead do we have to take it and send it down to the apex and then up from the apex? Why would we want to do that? What's the advantage of that? This may be one of the places where my schematic doesn't help us as much. So let's sneak back to the pretty picture from the textbook. When we're here in the pretty picture of the textbook, we again notice we can see all of these components. Our sinoatrial node, our atrioventricular node, the bundle, the atrioventricular bundle, which bundle of His leads into the bundle branches, leads into the Purkinje fibers. Right? Notice one thing about these Purkinje fibers is they feed into the papillary muscles to cause them to contract, to pull on the chordae tendineae. But here, this reminds us that with our ventricles, I draw the schematic, but remember, the great vessels, the arteries that are going to receive the blood are up here, superior, right? So notice there's an advantage to bringing the electrical signal down to the apex and then spreading it up. It gives us a directionality to the contraction, right? How do you squeeze a tube of toothpaste? Wait, let me rephrase that. How are you supposed to squeeze a tube of toothpaste?
On the
All right.
bottom.
From the bottom. You squeeze it from the bottom and you get a much more efficient. Now, I know everybody just grabs it in the middle and doesn't do that, but when it gets low, then you start to squeeze it from the bottom and squeeze it up because you get much more efficient with that directional contraction. And that's what's happening here. By bringing the signal down to the apex, we get a directional contraction of that ventricle. And as you get that directional contraction, that is going to more efficiently pump the blood out of the ventricles. All right. So let's do this again. Here in the armpit, we have our sinoatrial node. Again, it generates the first action potential. It's the pacemaker cells. And when it produces that action potential spreads along all of these contractile cells of the atria. And as it spreads across, and again, let's be more specific, the contractile cells as it spreads all across the contractile cells of the atria, what happens? Not a trick question. When you stimulate a contractile cell, what does that contractile cell do?
Contracts.
Contracts, absolutely. However, here's the one catch on this. As we know, the second we produce an action potential in a muscle cell, does it instantly start producing tension? Well, think back to your twitch. Is the first thing that happens when we stimulate it with a muscle action potential, the tension immediately starts going up?
No.
No. So one of the important things to remember about this process, an important thing to remember is that there is a delay between the excitation of a contractile cell and the actual contraction. All right. So the second a contractile cell gets stimulated, it isn't instantly generating tension. There is a bit of a delay. We have that latent period. So it's not a direct measure of the action, but it is a direct measure of the electrical activity. The cell gets stimulated, and we know when it gets stimulated, it's going to contract. Not instantly, there is a little bit of a delay. So we know this action potential is going to spread across the atria, and that spread across the atria is going to stimulate those contractile cells, and the atria is going to contract. All right. Now, as it's spreading across the atria, it can't get to the ventricles because of that pesky fibrous skeleton, but it can get to the atrioventricular node. And as we said, that atrioventricular node has very small, very thin cells. And what's the advantage of that?
It's more resistance.
It adds more resistance, absolutely. It increases the resistance, which decreases the flow, which delays the spread. So this delays the spread of the action potential. However, eventually, it's going to get into the bundle of His and the bundle branches, traveling down to the apex of the heart. And again, why do we want to bring it down to the apex of the heart before we bring it up the ventricles? To produce that directional contraction, absolutely, to make a more efficient contraction from that. But you guys have the right idea. Those Purkinje fibers are going to then directionally stimulate the ventricles to give us a directional contraction. And our ventricles contract. Remember, as we mentioned, our papillary muscles are going to contract first so that they can pull on those chordae tendinea to help to keep the cusps of our atrioventricular valves from prolapsing. And we pump the blood out. All right. Questions on that? All right, excellent. So we have this pretty picture from your textbook that does a great job of showing the steps in this process of how our electrical signal spreads across the heart. And we know the spread of that electrical signal is going to lead to the contraction. It's not a measurement of the contraction, but it leads to it. Oh, I'm sorry. Oh, great question. No, the two beats that you hear of the heartbeat are actually the closing of the valves. First, the closing of the atrioventricular valves, and then the closing of the semilunar valves. So you're not actually hearing the pumping, the changing of the volume. You're actually hearing the closing of the valves. Sorry, I didn't catch that question. Were there any others that I missed? Looks like I got all of them. I'm sorry. Thank you, Alex. Thank you for catching that. I didn't catch that there was a question there. Thank you for that. I appreciate it. You may misinterpret the question, but you let me see that there was one. So I do appreciate that. Excellent. Now, as I mentioned, this is the movement of electrical activity. We know it is going to produce the contraction, but it's not a direct measure of it. But it still would be really nice if we could actually see and measure the movement of electrical activity. But this is all going on inside of our body, and there's no way for us to be able to measure the movement of electrical activity that's going on inside of the body, is there? Is there a way to measure the movement? There is. Well, I know if, for instance, if I have a wiring problem in my house, I can get an electrician over here, and he has a fancy tool that he can hold up to the walls, and through the walls, he can tell when electricity is moving through the wires. But they can't do that on the human body, can they? Ashley seems to think that we can.
Isn't the EKG?
Yeah, exactly. That's exactly what an EKG is. An EKG or ECG, electrocardiogram, that is exactly what an electrocardiogram is. An electrocardiogram, an ECG, or an EKG, that's exactly what it is. It is the measure of the movement of electrical activity in the heart. And so we can externally actually measure these movements, quantify these movements. You're going to be doing it as part of your lab simulator, the interactive physiology, looking at that, and we will talk about that together as well. All right? Yes, one is German and one is English. That's the difference between ECG and an EKG. Yep, it's just how you spell it. All right? Excellent. Questions on that? All right, that is everything that I wanted to do for lecture today, but I do have two lab things I want to do. One of them, as you can see here, is to go a little bit more over the histology, and I want to spend a little bit more time, like we were talking about, going back over the blood vessels of the heart. Again, the nature of this class, because there is so much physiology to talk about, it's a little different from 430. In 430, we have more time to focus on the anatomy, and I can hold your hand and walk you through those things, and we go through all of it together. At this point, you should be sophisticated enough as a student to be able to successfully maneuver the anatomy on your own. However, there are some asynchronies, there are some asymmetries, there are some unique characteristics. And so what it allows me to do is I can leave all the basic easy stuff for you, and we can spend a little bit more time focusing on some of the more esoteric anatomical features. And so that's what I want to do. And since we just talked about the heart today, I want to go back to the heart and talk more about those blood vessels, because again, that's one of the big places where the anatomy, we keep drawing as this circle divided into 4 parts, but that's not really what it looks like. Even the picture that we see here, we know there's the circular left ventricle and the right one wraps around. It has a unique shape and it has a unique blood supply. And so I want to talk about that as well. So there are 2 things that I want to do, but I've talked a long time, so we'll take one more quick break, and then when we come back from this break, that will give us, we'll switch gears into lab mode. So it's 3:30 right now. Let's come back. Let's just take a quick 10-minute break to make sure that we get through this. Just a chance to stretch. Come back. Actually, it's 3:32. Let's come back at 3:42, and at 3:42, we will pick up from here. So we will restart, and we will go into the lab at 3:42. All right, questions on any of that? All right, see you in 10 minutes. So, as I mentioned, what I want to finish off with today is some of the stuff that we have already talked about, but I want to make sure to emphasize it. Starting first with the histology. This histology shouldn't be too new to you. Again, you should have been responsible for cardiac muscle when you were in 430. So here, for instance, we see some of those branching cells. Again, I quickly stole some of these pictures from the interweb, so some of them are already labeled. We can see here we're showing nuclei, and here's some examples of the intercalated discs in between them and some of the connective tissue in between them. So all of that is new. And again, lots of examples. I like two things about this picture. I like, again, that you can see the striations really nicely, but this one really does a good job of showing all of the space in between. And that's these big nuclei that you're seeing. Most of these are the nuclei of the connective tissue, forming that fibrous skeleton. So that is the structure that is being formed by this fibrous connective tissue that is found in here. And we see a really cool example of an intercalated disc there. And remember, these are what are holding the cells together. And also, as we talked about, have massive number of desmosomes, massive number of gap junctions in them. And just another, again, different stains show this different ways. And again, this is another one that does an amazing job of showing us some of that connective tissue in between. And notice, again, every once in a while in all the tissues we see big round cells that are totally clear like that. What do you think that is?
Fat?
Yeah, or more specifically an adipocyte, right? This is an adipocyte. This is one of those fat cells. Absolutely. I make a point of emphasizing this because, as we've also often talked about, you find fat pretty much everywhere. However, now we're more sophisticated. Now, as we look at this tissue here, we see that all these cells have striations. They have those intercalated discs, and those striations mean they have myofibrils. And those myofibrils mean that they have sarcomeres. And those sarcomeres means that these are going to contract. Notice every cell we've looked at so far is part of that 99%ers, those contractile cells, the ones that are generating the force, right? What we can't see, at least not in this picture, are the 1%ers, those conductive cells. And as it turns out, the easiest place to see that is in the ventricles. Remember, as we talked about in the ventricles, we have large numbers of ththose Purkinje fibers that are spreading out throughout the surface of, again, remember, it's still part of the myocardium. And that's what we see here. I couldn't find a picture that wasn't labeled that I didn't use for the exam already. So I've got this one here that happens to be labeled. But what I want you to see is here we see some of the fascicles of our myocardium. And here we see the connective tissue forming some of the septums. We see some blood vessels in here. But here, a part of the myocardium. Notice it is close to the endocardium. It is often found close to the endocardium. Remember, the endocardium is just the simple squamous epithelial tissue and a little bit of areolar connective tissue that is that endocardium. So here we have these cells, and these are the conductive cells. Now, notice when we look at them at first, they almost look a little bit like adipocytes. They're bigger and they're more pillowy in appearance. They definitely don't have striations, right? They definitely don't have myofibrils or sarcomeres or any of those things inside of them. However, notice when we see a nucleus inside it, the nucleus is right in the middle. Is that what we see in adipocytes? Do adipocytes have their nucleus right in the middle? No, they're shoved way to the side by that clear vesicle of that oil droplet. And notice, while these are big and pillowy, they're not clear. They are pillowy, but they're not clear in the center. They have a centrally located nucleus. So if we were just whipping through this and hadn't paid any attention, we would have just thought that that was a fat deposit. But now that we're looking at it more carefully, we can see that these clusters of cells that are part of the myocardium are actually examples of these Purkinje fibers found here in the myocardium, and those are part of the 1%ers. Those are part of those conductive cells. And also notice the magnification we're looking at here, right? We can barely make out the size of the contractile cells here, but we can clearly make out the size of those conductive cells. So they're big, pillowy, centrally located nucleus within the myofibril, often clustered together like this. And those are things that are going to be a dead giveaway when you look at this histologically. And you're tested on this on the exam, because this is one of the things you're responsible for histologically. All right?
Please say it again, where they are located, usually.
In the myocardium, often, not always, sometimes you'll see them over here, but often you will see them close to the endocardium as well. All right? Excellent. Is there anything else on our histology list for this? Again, not including arteries and veins. We'll talk about the arteries and veins when we get to that. But for the cardiac muscle, I think that's everything for those. Ashley, are you telling me no, that I've covered everything? Or no, that I'm missing something? Or was that no to something else? Anybody have the histology list printed out, looked at it yet? For shame. All right, excellent. All right, well, let's pretend that's everything on the list. And if not, you can ask me later. All right, excellent. Let's switch from that then. To this, our vascular system. Again, we have been so far just talking about the heart, so let's remind ourselves of some of the things that we have been discussing. Our oxygen-rich blood leaves the left ventricle, and when it leaves the left ventricle, what structure does it pass through?
Like the valve, or?
Yeah, what structure does it pass through? What valve does it pass through?
Aortic valve.
Okay, aortic valve is good, although could we be more descriptive?
Yeah, I don't know what it's called though.
Aortic semilunar valve.
Ah, okay.
All right, so we have that aortic semilunar valve that it is going to pass through. And you're absolutely correct. From the aortic semilunar valve, what blood vessel does it feed into?
Aorta.
Although, what do we want to say? Ascending aorta. We want to say the ascending aorta. Remember, one of the reasons we're dividing the aorta up into bits is that the different bits distribute blood to different parts of the body. And here is a great example. As we already talked about earlier today, how many blood vessels come off of the ascending aorta?
3.
Nope. The ascending aorta has how many?
2.
2. Just 2. Absolutely. And what did we say those were again?
The coronary arteries.
There you go. Right? The coronary arteries, the right and the left coronary arteries. All right, let's look at a pretty picture. And here, when we look at the pretty picture, nice and big, we can see this a little bit more clearly. Notice again, as we talked about, and let's start over here. Here is the left coronary artery. And remember, as we talked about, it is typically hidden by the pulmonary trunk. This is the pulmonary trunk coming out of the right ventricle, and then it branches into the pulmonary arteries. So by removing it, we're able to see the left coronary artery, right? Remember, we're talking about some big asymmetries. So let's do this. Notice this is it. This part, that didn't show up well at all. So let's try green. Will green show up better? I don't like that one. How about a darker green? There you go. That's pretty good. That's it right there. That right there is the extent of the left coronary artery. It's tiny. It's shy. It hides behind the pulmonary trunk. So typically, if the pulmonary trunk is here, you basically just see the head of it coming out right before it splits. Whereas notice the right coronary artery comes off the ascending aorta. Remember, in that atrioventricular sulcus, goes all the way along the edge of this and goes onto the posterior side. All right. Now notice there are a lot of branches. There are a lot of anastomoses. That's a fun word we haven't talked about yet. Anyone know what an anastomosis is? No. An anastomosis is an elaborate network. One of the things we want to do, if you think of it this way, for this part of the heart right here, do we necessarily want to have just one way for blood to get to that particular cell?
Yes.
Well, if there's just one way for blood to get there, and that blood passageway gets blocked just for 6 seconds, let's say, is that going to be that big of a deal to that one cell?
Yes.
Possibly,
Yes.
depending on what's going on. Absolutely, it could be damaged. So by having multiple routes, and that's what anastomoses. Anastomoses are these elaborate networks of vessels. They can lead a lot of branches. So there's all these arterial branches that are going off to different areas and interconnecting with each other. Here we see a big interconnection down here. So there's all sorts of messy stuff that is going on. But along the way, occasionally some large branches come off. And notice one of those large branches that come off, and I guess I have to use the word large. These pictures are actually straight out of your textbook. And some of them might be some of them might also be the ones that are unlabeled in your, in the module. Ashley's nodding, so there you go. See, there you go, perfect, excellent. Those are the unlabeled pictures that are on Canvas. Perfect, excellent. Notice here along the lateral aspect of the right ventricle, the lateral aspect is the marginal artery, right along the margin of the edge, right along the edge. So right along the margin of the right ventricle is where that marginal artery is located. So notice our right coronary artery feeds into the marginal artery. But notice also, as we talked about, and I think this does it, here we go, we see it on the back. Notice that right coronary artery continues all the way around onto the back side of the heart, where it then branches and goes down in that indentation between the ventricles on the back of the heart. And what do you think we might call that indentation between the ventricles on the back of the heart? You'll get a partial credit for sulcus, but we want to give the entire name. There you go, posterior interventricular sulcus. Excellent. And guess what artery that right coronary artery forms that goes into the posterior interventricular sulcus?
Posterior interventricular artery.
Perfect. It becomes the posterior interventricular artery. Excellent. Bing. So notice our right coronary artery, very, very different path from our left coronary artery. It wraps all the way around the right side of the heart. Notice again on that lateral edge, it helps to form branches off the marginal artery, comes onto the back and down the center of the back of the heart in that posterior interventricular sulcus, forming the posterior interventricular artery. All right. So questions on the path of the right coronary artery? All right, excellent. Then let's go back to the front and see what happens with that left coronary artery. Remember, as we mentioned, it is much, much smaller, just basically hiding behind the pulmonary trunk, and then it branches. And as we talked about earlier, one of those branches goes right down the front of the heart in that indentation between the ventricles on the front of the heart. And what did we call that indentation on the front of the heart again?
Anterior interventricular sulcus.
Notice the interventricular sulci, anterior and posterior, are the indentations, and the septum is in between, right? Let's take a transverse section through the heart. If we were to take a or really through the ventricles, If we were to take a transverse section through the ventricles, again, there's that little bit of indentation between the ventricles on the front. On the back, there's a little bit of an indentation, and again, obviously I'm exaggerating this for effect. There's the posterior interventricular sulcus, the anterior interventricular sulcus, and between them is the big wall, the septum, that divides the two ventricles in half. So again, septum and sulcus are not the same thing. The interventricular septum is the muscular wall between the ventricles. The interventricular sulci, anterior and posterior, are the indentations on the surface.
Yes.
All right, excellent. And as was pointed out, what sits in the anterior interventricular sulcus? The anterior interventricular artery. All right. Now notice the second branch that comes off of the left coronary artery sits in the atrioventricular sulcus and wraps around the heart on the left side. Or you could say it wraps around the circumference of the heart on the left side, which helps you to remember that it is the circumflex artery. And notice that circumflex artery, as we clear and flip it over, circumflex artery basically comes all the way down to the back, which feeds a second marginal artery along the edge of the left side of the heart. So notice we have two marginal arteries, one on the left, one on the right. And so in this fashion, notice we've essentially been able to bring blood to all parts of the heart with these big, large blood vessels. And they've been very convenient. The blood vessel in the posterior interventricular sulcus is the posterior interventricular artery. The blood vessel in the anterior interventricular sulcus is the anterior interventricular artery. Right? We have marginal arteries, circumflex arteries. So are we going to have those exact same names for the veins as well?
No.
No. Again, for whatever reason, they've given it, because of the asymmetries, they've given us different names. So again, remember the capillaries are going to be microscopic. They're going to be all over the place to make sure that every single cell of this heart is getting the oxygen and the nutrients that it needs. Alex, I won't make you specify the marginal arteries for that. Again, I've just put it on there as marginal. No, there are some on both sides, both the front and the left. And then we'll see the blood vessels that go along with them as well, the veins that go along with them. That's really the easiest way you're going to distinguish them. All right. All right, because notice even here that they have right coronary artery, but they don't have right marginal or left marginal. They're just really both called marginal arteries. Again, the only reason I can think of is why is because anatomists hate you.
I have a question.
Yes.
In the book, it says that the right coronary artery sits in the coronary sulcus. So do you want us to call it the atrioventricular sulcus?
No. So great. That's an excellent question. And my answer to that is you are always welcome to use any appropriate anatomical term. So yes, coronary sulcus is a perfectly acceptable anatomical term, and so that is perfectly acceptable as well. Same way, bundle of His and atrioventricular bundle are both anatomically appropriate terms, and so both can be used. So yes, there oftentimes things have multiple names, and anytime you do that, you are welcome to use any name as long as it is appropriate and accurate. All right, excellent.
I have a question.
Yes.
So the left coronary artery branches into circumflex artery and anterior interventricular artery?
Correct.
Okay.
And it terminates at that point. It doesn't continue on, right? Notice the right coronary artery, when it branches to form the marginal artery, it keeps going. but that's not the case with the left coronary. The left coronary terminates, and when it terminates, then it forms the circumflex and the anterior interventricular arteries.
Okay.
Okay? Excellent. All right, we have made it around the arteries. Let's identify the veins. And like I said, it would make all the sense in the world that this big, large vein located in the indentation on the anterior side of the heart should be the anterior interventricular vein, but it's not. Instead, it's called the great cardiac vein. And it's called the great cardiac vein because it has to travel the longest distance, right? It is the longest of our cardiac veins, and so that is why it is called the great cardiac vein. It starts here on the anterior interventricular sulcus, and then it goes and follows along with the circumflex artery around to the posterior side of the heart. All right?
But from the posterior side, it calls middle cardiac vein, right?
Nope, middle cardiac vein is something different. This goes all the way around to the back, and we'll see that in just a moment.
Okay.
Before we do that, the widest we'll talk about in a second as well. We got to see the back of the heart for that as well, Yvonne. All right. Now notice, remember, over here we had a marginal artery, and right next to it, we have the small cardiac vein. This illustration, and again, remember, this isn't 100% what it looks like. This is an artistic representation of what it looks like. Often these are side by side, right on top of each other on that lateral aspect, and that's how I always remember it, right? If you try to marginalize someone, like say, for instance, there happens to be a professional athlete who is tired of the social injustice and says that I am not going to play basketball tonight until something better happens, and someone else on Twitter gets on there and tells them that they should go to China, then that would be an example of trying to marginalize someone. And if you're trying to marginalize someone, you're trying to make someone feel small. And so lo and behold, right there where our marginal artery is where our small cardiac vein would be located. All right, and it happens to be one of the smallest of our cardiac veins, at least the smallest one that you are responsible for. And it needs to travel around to the posterior side as well. But because it's on the edge, it doesn't have as far to go. So from there, let's switch to the posterior view. Here on the posterior view, and I'll use different colors this time to make sure we understand this. Here is that great cardiac vein coming around from the anterior side. Here, we'll use yellow, is that small cardiac vein that started on the edge and came up from here. And as someone pointed out, we have a middle cardiac vein. That middle cardiac vein should be called the posterior interventricular vein, but it's not. And since it's not the biggest and it's not the smallest, it's the middlest. So it is the middle cardiac vein. However, remember also that our left ventricle has a much thicker muscular wall because it has to generate much more power. And so it's not surprising that it needs more blood. And if it needs more blood, it also has to drain more blood. So notice we have a fourth vein, and that fourth vein is known as the posterior cardiac vein. However, again, we were just talking about different appropriate anatomical terms. This is sometimes actually referred to as the posterior cardiac vein of the left ventricle. Now, you don't have to use all of that in defining it. You can just say posterior cardiac vein, but as you're reading the materials, you may sometimes turn into that term as well. It's the posterior cardiac vein of the left ventricle, or just the posterior cardiac vein is fine. Now notice, all four of these veins' jobs are to drain blood from the heart. We need to get a lot of oxygen-rich blood to the heart, and we need to get rid of that oxygen-poor blood as easily as possible. Towards that goal, then, we need a nice, big, loose blood vessel. Our widest of blood vessels, nice, big, loose blood vessel that can easily collect all of the blood coming back from the heart. And what is that nice, big, loose blood vessel located here on the back side of the heart where we collect all of the blood from the heart?
Coronary sinus.
Yeah, if you're not sure, you can follow the leader up here and see, it's the coronary sinus. Absolutely. This is the coronary sinus. This coronary sinus is this big, large, loose blood vessel. Remember, a sinus is a big, large, loose vein. So yes, this is the widest vein in this because it's big and loose. It's big open space. And notice, and I think I have to decrease the size of this. If you look closely, and I'll remove my highlighting in just a second so you can see it, but you can actually notice where it actually feeds the blood into the right atrium. Notice here is the inferior vena cava, here is the superior vena cava, and here is the coronary sinus. So I'm going to go ahead and clear my pictures here so we can see this more clearly. Coronary sinus, inferior vena cava, superior vena cava, all of these feed into the right atrium. Okay, need to know it. Direction it carries the blood to the heart from the heart, that works. Condition of the blood, oxygen rich, oxygen poor, that works. What they feed into, what they feed out of, yeah, for the most part that works. I mean, I wouldn't say what does the posterior interventricular artery feed its blood into, because there aren't any named blood vessels on your list that do that. But do you need to know that it gets its blood from the right coronary artery? Sure, I think that relationship would work. Yeah. So understanding the blood flow through the heart is definitely something that I think is reasonable. Not every single one of those 5 questions will be appropriate for all of them, but for most of them they would work. Yeah.
So we give you some ideas.
Oh, well, again, yeah. One of the things that I don't like about this testing format is I like controlling my exams. I like having the right number of hard questions and easy questions, and I've got lots of different questions that I have that I can rifle through and find just the right mix that I think really does a good job of giving an even distribution over all of the important material. The problem with this online format is every single one of you is going to get a unique exam on the first test, right? Okay. Our lab exam, if you think about it, our lab exam, basically, if you notice, and again, let's just throw random numbers out. Let's say that I have 50 blood vessels on that list. I don't know if that's high or low. I haven't bothered counting. But if you think about it, with 50 blood vessels on that list, and each one of them has 5 possible questions, that's 250 questions just from the blood vessels. That doesn't include blood, that doesn't include the anatomy of the arteries and veins, that doesn't include the heart, that doesn't include any of those things. It's 250, and guess what? All 250 of those questions are in the question bank. All right, I think for this exam, for the lab exam, I think I have over 500 questions in that question bank. That is going to be, now you're not going to get 500 of them. You're only going to get about 430. So it's not going to be that bad. No, my guess is, and again, I'm still working on these banks. No, my guess, this test usually is 80, 85, somewhere around that range. So it'll be somewhere around that range, the number of questions that it will be. And it's the same way with the essay questions. You know, every possible essay question I can think of has been thrown, or that I have in my bank has been thrown in there, and everybody's going to get a different one. So some people may get, 3 hard questions. Somebody else might only get 2 hard questions. The hope is that over a semester, the randomness of that evens out. right? So your first test may be a little bit easier, but your second test may be a little bit harder. Somebody else's first test may be a little bit harder, but their second test is going to be a little bit easier. The hope is that it evens out. Is it possible that you could get a hard test for the first and the second, or an easy test for the first and the second? Yes, but hopefully over the 11 exams you have for this class, it will even itself out enough that everybody is being tested equally on this. But unfortunately, it's something I can't control. So yeah, all of these potential questions are already in there. Everything I could possibly think of, I am throwing in or still throwing in. I'm still working on these questions. So again, you're not giving me any new ideas. I've been working for months on these ideas to try to get them in.
I was kidding.
No, but you're right though. There are in a normal classroom, there are often times where something comes up and I'm like, oh, that's a great question. I'll add that in there. But in the next test or something like that, because it's important and relevant. But I can't do that in this, because even if I throw a new question in there, there's no guarantee that all of you are going to get it, right? Because it's randomly picking all the questions. So I do still add, I do still modify, I do still keep thinking of things, but it's not the same as doing it in person that way. So it's weird and I don't like it, but again, it is what it is. Yes, there are time limits for both the lab and lecture exams. Absolutely. Yep, so both the lab and lecture exams. The lab exam is typically 1 minute per question with about 10 or 15 minutes extra thrown in. That gives you your scanning time. That gives you a little bit of critical thinking time, things along those lines. But again, lab exams should be pretty bang, 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 bang. Essay questions, I appreciate that people vary dramatically on how they tackle it. Some people really like to work it out first. And again, that's why you have that whiteboard. If you need to use that whiteboard as scratch paper to write on in there. Or again, the nice thing about this essay format is you can actually do your brain dump into the question and then underneath it write it and then delete what you didn't use or something along those lines as you're going through it that way. And so I try to be a little bit more flexible with the time for the lecture exams. So the time's a little tighter for the lab exams, but it's a little more lenient for the lecture exam. But remember, all of it has to fit within the class time. That's one of the big differences for those that were in the spring versus now. Because we were being forced online and all the craziness of it, we were much more flexible with the timing of the exams. What I want to remind you is you are fully aware, 100% aware of the fact that you are taking a synchronous online class. And so as such, you can take the lab exam first or lecture exam first. You can do it in any order that you want, but both exams have to be completed between our class time, between noon and 4:30. All right?
I have a question.
Yes.
Do you grade the essay questions yourself, or does like a computer do it somehow?
It's me. It's all me. Yes.
Okay.
It takes me about a week to grade the exams under the best of circumstances. I will do the best that I can. But yes, I do all the grading. I don't have TAs. Our instructor assistants have offered, but for me, it is important that the grading be standard so that I use the same rules for everything. And so I can't let somebody else grade a third of them and I grade the other 2/3 because I can't guarantee that the standards will be the same on that. So yes, I read every single essay question. So again, as horrible as you feel while you're sitting there taking it, just imagine what it's like for me having to read 33 of them. All right. So again, it's definitely, but yes, it's all me. All right. Excellent. So there is our heart and the blood vessels associated with that. We did a histology. We've talked about this. On Tuesday, we will pick up with the ECG and talk about the movement of that. Where's my list? So on Tuesday, okay, excellent, perfect. We'll get a chance to talk about it on Tuesday, and then on Thursday, your interactive physiology on the ECG is going to be due as well. So we'll get a chance to talk about it first, discuss it, and then you'll get to do that great handout from the interactive physiology. And hopefully while you're on there, you'll look at other interactive physiology stuff as well. That's really why I'm giving that assignment. It's the worksheet is pretty basic, but it's an excuse to encourage you to get on there and see that there is an interactive physiology. So hopefully you will use the other resources that are there. All right. Excellent. Questions on any of that? I did warn you on Tuesday we wouldn't get off early all the time. So I'm still not, I didn't keep you all the way to 4:35. So you are getting a little bit of extra time. But any questions on that? That's everything that I wanted to cover for today. All right, excellent. You guys have a very good weekend. Be healthy, be happy, be healthy, be safe. Real quickly, as always, as a reminder, listen to your scientists, listen to the doctors, do not listen to the politicians. All right. If you have not done so already, register to vote. I will not tell you who to vote for, but I will tell you, you need to get out and vote, right? All of you are old enough. This is your civic duty. Get out there, be civic, make the wise, responsible choices, right? And do what is right. You have to be a part of this process. All right? All right. Be happy, be healthy, be safe. I'll see you guys. Have a good weekend. See you on Tuesday.
Bye.
Bye.
Thank you, Dr. Slutsky.
Thank you.